that we found, and I will welcome to all of you for the two-day international seminar on recent microbial advancement and impact of chemical pollutants on the components of ecosystem, that is RMACPE 2025. This is the technical session 2 that we are organizing in the day 2, and I would like to welcome the chairperson of the technical session, Dr. K. Patel sir, Assistant and Head of Department of Microbiology and Department of Biotechnology, Government PCL PC College. He is having the teaching experience of more than 13 years in higher education, and he is having more than 7 years of teaching experience in medical education also. Sir is having more than 47 national and international research paper publication. I would like to welcome you, sir, for this technical session as a chairperson. And
thank you, madam.
And I would also like to welcome the co-chairperson of the technical session. She is Dr. Smriti Pandey, Assistant Professor and Head Department of Microbiology, CMT PG College, Bilaspur, Chhattisgarh. Ma'am is having more than 12 years of teaching experience in UG and PG classes. Along with, she is having many research paper, patent, book and book chapters along with the national and international conferences. So I am feeling very lucky to have both the chairperson and co-chairperson here, sir and ma'am. I would like to again welcome both of you to this technical session.
Who is the first participant? Akansha Sharma, Aarti Shahu, Akhilesh Pandey. Who is the first? Aman.
Whosoever available can join first. We will write down the name and start the technical session.
Good afternoon, all respected members. Myself, Dr. Vaishali Pimple. So, can I start with my presentation?
Please turn on your video. Please turn on your video.
Dr. Vaishali
Pimple.
Dr. Vaishali Pimple.
I'm Assistant Professor in Department of Microbiology, Dharampet MP Dev Memorial Science College, Nagpur.
Okay.
Okay.
Okay, please continue.
Yes, sir. First of all, I would like to thank D.L.S College for providing this opportunity and organizing international seminar in collaboration with the Microbiology Society of India. So, with the permission of respected members, can I switch off my camera and start sharing my screen?
Madam, please continue your camera and screen also. Your time is 5 to 7 minutes, please.
Okay, sir. So, here message is appearing that request has been sent.
Request accepted.
Please accept it.
Yeah.
Okay. So, is my presentation visible?
Yes.
Ma'am, you are not audible. Please unmute.
Ma'am, you are not audible. Miss Anjana Khare, please unmute yourself.
Am I audible, sir?
Yes, now you are audible.
Yeah, thank you so much. So we are living in the era of antibacterial or antibiotic resistance, and we are always in a search of the substitutes for antibiotics. So plant essential oils are one of the substitute for antibiotics, because it contains large constituents like p-cymine, limonene, menthol, which have demonstrated potential antibacterial, antiseptic, antiviral, and antioxidant activity. So, mostly essential oils are used in agriculture and pharmaceutical and sanitary products, but these have some limitations as a use as antibacterial agents because of their low stability, strong organoleptic properties, and low stability. So here in my research, we have found one substitute, or we can say one of the option as a nano encapsulation technology for synthesis of essential oil based novel antimicrobial agents. So, which can be in the form of nanoemulsions, liposomes, solid lipid nanoparticles, and polymeric nanoparticles. So, we can organize these nanocapsules or nanospheres or nanoemulsions. And what are actually, in my research, I have synthesized some nanoemulsions of plant essential oils. Switching to the my next, what are actually the nanoemulsions? So antimicrobial nanoemulsions are oil in water nanoemulsions with a droplet size in the range of 200 to 600 nanometer, and are stabilized by the surfactants and demonstrate potential uh, broad spectrum activity against number of pathogens. So here we have tested the prepared the synthesized various nanoemulsions from oils, and tested it against the clinical isolates of Asorius, E. coli, and Salmonella typhi, which are procured from the Vishakha Pathology Laboratory of Nagpur. And the various plant essential oils which are used for synthesis were tea tree oil, thyme oil, clove oil, and cinnamon bark oil. Coming to the methodology, how I have prepared these nanoemulsions, which are I prepared it as per the directions or methodology used by Chandrasekhar et al. We have used tween 80 and tween 80 and tween 20 as a surfactant and mixed it with the ratio of 1 is to 1, 1 is to 2, 1 is to 3, 1 is to 4, volume by volume, and mixed it with stirring at 650 rpm for 30 minutes. And during this process, deionized water is added to the that particular crude mixture, uh, and along with the surfactant under study. And this mixture along with the surfactant was then ultrasonicated with the probe ultrasonicator of 120 watts and 30 kilohertz frequency for 70 minutes, which resulted into the formulation of nanoemulsions, which are then further characterized for their physicochemical parameters and stability studies. So this is the chart for the composition of the nanoemulsions, the ratio of oil and surfactant concentration, and the quantity of essential oil and surfactant and deionized water used for the preparation of essential oils. So these are the various essential oil nanoemulsions of the cinnamon oil, thyme oil, clove oil, and the tea tree oil used by me. So for characterization, we have done various physicochemical parameters, heating cooling cycle, measurement of droplet size. So this is a chart for the physicochemical parameters or physicochemical characterization of nanoemulsions. Coming to the main point, in our research, we have found that tea tree oil with the composition of the and thyme oil with the between 80 have given the maximum antibacterial activity. Then cinnamon oil nanoemulsion in 1 is to 4 proportion with twin 20 have given maximum antibacterial activity, and therefore we have done the particle or the nanoemulsion size, droplet size estimation, and polydispersibility index study of these nanoemulsions, which were selected for the antibacterial activity.
Dr.
So
Vaishali, summarize.
we have found that for the yeah. So this is the data Then we have combined the various nano emulsions, and we have estimated the data using post hoc Tukey's test. And this is the data. These are the findings of antibacterial activity of the combinations of various nano emulsions. Against the Staphylococcus aureus, you can easily observe the zone of inhibitions which are observed in the combination of nano emulsions. And if we have interpreted that, we can see that these antibacterial nanoemulsions are showing maximum antibacterial activity. But if we use in the combination these nanoemulsions, like if we mix these two nanoemulsions, like the cinnamon oil nanoemulsions and the tea tree oil nanoemulsions and the various nanoemulsions, the antibacterial activity of these nanoemulsion combinations gets enhanced. So this graph represent the antibacterial activity of individual nanoemulsions in arrow, and the bars represent the antibacterial activity of the combination of nanoemulsions. So these are the results. These are the results for E. coli, and
Dr. Vishal.
yes, and these are the changes which are obtained in the bacterial structure. We have studied it for the electron microscopy at the Raska laboratories in the Hyderabad also. So when on Staphylococcus we have, so when we treat the nanoemulsion with the SRES, we have seen that leakage of nuclear material occurs, disintegration of the bacterial cell membrane occurs, and the bacterial cell septums also get disturbed. So we coming to the conclusion that tea tree oil, then clove oil, and the cinnamon oil, and these oils, nanoemulsions possess potential antibacterial activity, and these antibacterial activities can be enhanced by using nanoemulsions, and which can be further augmented by using the combination of nanoemulsions, which can be proven with electron microscopic studies. So these are the future prospects. I have patented these two formulations, Twin 20 and Twin 80. That is cinnamon oil nanoemulsions I have patented. Then I have patented the methodology of the preparation of nanoemulsions. So on this topic, I have obtained 2 international patents, sir. Thank you so much, sir.
Okay, thank you. Dr. Vaishali. हाँ, Dr. Vaishali.
Yes, sir.
हाँ, आपने जो अपना आइडेंटिफिकेशन किया है बैक्टीरिया का, ये डॉक्टर यज्ञेश ठक्कर के यहाँ
Yes, sir.
किया है,
हाँ.
डॉक्टर यज्ञेश ठक्कर, और क्या बोलते हैं या स्टेफिलोकोकस और ये आपने क्या ये मारे से किया नहीं किया था?
सर MRSA पे मेरा स्टडीज अभी चल रहा है. मेरा प्रोजेक्ट है यूनिवर्सिटी में, मैंने डाला है, वो सैंक्शन हो चुका है. तो MRSA पे हम लोग कर रहे हैं, और उसका सिल्वर नैनो पार्टिकल्स के साथ भी हम कर रहे हैं, और हम एक वुंड हीलिंग मटेरियल बनाने की कोशिश कर रहे हैं.
ठीक है, चलिए, थैंक यू.
यस सर.
मैडम
थैंक यू.
क्वेश्चन कर रही हैं. चलिए, ठीक है, नेक्स्ट.
थैंक यू सो मच सर.
Thank you very much. Now, next participants. अंकित कुमार भागवत, आकांक्षा, आरती साहू, अभिलाष पांडे, अभिषेक सिंह सिद्धा, अमन, whosoever present for oral presentation can start their presentation. आनंद कुमार, अनीता पांडे, अनुपमा सिप्पी, अर्घ्यदीप, बलराम कुर्रे, दीप्ति गुप्ता, देव नारायण पटेल, Dhaneshwar Prasad. I can see Balram Kurre here. Can you please start? Next, Dolamani Sahu, Dr. Alka Dubey, Dr. Prabha Raj, Dr. P.K. Singh Rana, Dr. Deepika, Dr. Monica, Dr. R.P. Sharma, Arghyadeep Bhattacharya, Anjana Khare Gupta, Harjit Singh, Hina Kausar.
Hello, yes, ma'am. This is Harjit Singh.
Okay, sir, you can start.
हरजीत, पूरा नाम क्या है आपका?
Harjit Singh.
जी हरजीत सिंह बस है सर।
हरजीत सिंह। सीरियल नंबर क्या है?
29.
29। हाँ, हरजीत, हरजीत।
यस सर।
हाँ, आपको 5 मिनट का समय है, फटाफट प्रेजेंट कीजिए।
जी जी जी। Good afternoon to respected chairperson Dr. K.K. Patel sir and co-chairperson Dr. Smriti Pandey madam. Opportunity to present my idea.
हरजीत, थोड़ा जोर से बोलो, स्पीक लाउडली, फास्ट।
Yes, sir. Yes, sir. This is my topic: screening for the production and correction D.L.S from endophytic fungi. Endophytes are the microorganisms that reside inside the plant tissues without causing any harm. Symptoms to the plant such as Fusarium roseum, Bread Rhizobium japonicum, L.S. which is the enzyme which catalyzes the amide bond in the L.S. to aspartic acid and ammonia. There are some sources of L.S. such as bacteria, fungi, plant, and animals. L.S. are used as a drug for the treatment of acute lymphoblastic leukemia, and it is first time approved by FDA in 1978. Also, it is used as a food processing aid to reduce the formation of acrylamide, which is carcinogenic in food material. In these commonly used drugs are nowadays a lot of immunogenic complications, a lot of side effects. So I can decided to uh, demand for a new source and also efficiency and characterization of some novel therapeutic properties which is found in these endophytic fungi. These are the mechanism of action of L.S. leukemic cells are which are synthesized here are dependent on exogenous supply of L.S. and L.S. which catalyze the conversion of L.S. to aspartic acid ammonia. The enzyme cut off the supply of L.S. in blood and cancer cells die as they become unable to build their protein. This is the objective methods for isolation of endophytic fungi. Will be to be collected medicinal plants such as Asimum sanctum, Aja directa indica, aloe vera, Withania somniferea are the plants to be chosen for this research. For surface sterilization, we use sodium hypochlorite followed by 70% ethanol, and for their culture, potato dextrose agar media to be solid state fermentation will be performed by using orange peel as a substrate for low cost and all the chromatography method to be used, and then final when it will be isolate crude protein which can be then correct SDS base. These are the expected research. Currently, I study describe that beside bacteria, plant, and other parasitic fungi of the microorganisms which can be proved to be better source of less less side effect and with low cost substrate.
चलो कंटिन्यू करो और कुछ बचा हरजीत
No,
चलो
sir.
थैंक यू मैडम आपका क्वेश्चन प्लीज चलो
Harijit, remove your slide share.
Okay, thank you very much. Now next participants. Arghyadeep Bhattacharya.
Okay, sir.
Harijit, remove your slide. Okay.
Am I audible?
yes Arghyadeep Bhattacharya.
Yeah. So good
You are
afternoon,
from
organizers, my respected professors. Myself, Arghyadeep Bhattacharya, the research scholar of National Institute of Technology, Durgapur. So I am sharing my screen.
Arghyadeep.
Yes.
Your time is 5 minutes. Please
Okay.
continue.
So my screen is visible?
Visible.
Yes, visible.
Okay. Fantastic. Thank you. So today my point of talk is the bioremediation and bioabsorption of chromium by Brevibacillus brevis US 575 isolated from the tertiary effluents and their potential industrial application. So I am belong from the state of West Bengal, and in the state of West Bengal there are various tannery industries located in the South 24 Parganas, and from these tannery industries 2 lakh metric tons of leather are processed every year. Now, there has been excessive discharge of chromium sulfide and chloride, and chromium is one of the most toxic inorganic contaminants. Bioremediation offers the possibility of using living organisms to degrade and remove toxic environmental contaminants and to convert them from the more toxic to the less toxic form. So my investigation deals with this characterization of the ternary effluents and the ability of the microorganisms to biodegrade the components present in the effluent that is responsible for bioremediation and bioabsorption. So these are my objectives, and this is the schematical representation of my total work. So we have collected our samples from the Bantala leather complex in the district of South Chobbish Pargona. We have isolated and characterized the microbes. We have found initially 4 isolates. And we have found that isolate 1 has the highest chromium reduction potential. From the leather degradation assay, we can show that isolate 1 and isolate 4 shows the most highest leather degrading potential. Now, why we are so much interested on leather? Because this tannary effluents consist deteriorated leather particles in the effluent. Our isolates have enormous promising abilities to produce protease as well as to produce lipase, which are different industrial enzymes. These are the lipid and protein degradation assays, and these are the keratinase assay. We have shown that our isolate has the highest keratinase production at a pH of 8 and at a temperature of 40 degrees centigrade, which is very much optimum for keratinase production by our isolates. Now, owing to the multidimensional ability of these isolates, we have characterized and we have identified these isolates. That isolate 1 is the Bacillus cereus F481072, and the isolate 4 corresponds to Brevibacillus brevis US 575. Among these, we have proceed with Brevibacillus brevis with biosorption experiments. So these are the different experiments where we have optimized the different experimental parameters from initial metal ion concentration at 200 μg per ml at a time period of 60 minutes at a biomass concentration of 3 gram per liter at a pH of 2, at a temperature of 45 degrees centigrade, and at an agitation speed of 80 rpm, we have found the highest removal efficiency of chromium from these effluents. We have studied the adsorption isotherms. We have taken Langmuir model and the Frederick model. The capacity of the biosorbent to adsorb maximally was 108.58 as obtained from the Langmuir model. And we have shown that the R square is 0.94, but the Frederick model 0.98, and it shows that the mechanism of adsorption was of monolayer type. And it also been indicated that the 1 by n value is within 1, which is actually showing a favorable adsorption process. So this is the desorption study and the elution study. We have found the highest percentage of recovery with 1 molar hydrochloric acid at a time period of 60 minutes. We have also checked with other elements as well. We also study with HCl, EDTA, NOH and nitric acid, and among all the elements used, we have shown that 0. molar nitric acid proved to be the best element in our experiments. We have also performed the regeneration experiments with raw biomass and in the bacterial biomass. From the experiments, you can see that the elution of these chromium ions from these bacterial biomass beads is stable for the 3 cycles compared to the raw biomass. So, we can give a message to the society that these bacterial biomass beads could be regenerated for use in the removal of metal ions from a contaminated aqueous environment. And these two experiments are very much crucial in our study, the desorption experiments as well as the elution experiments, as well as to make this total procedure cost competitive. We can perform this regeneration experiments is showing that the bacterial biomass of our isolate Brevibacillus brevis US 575 could be used, could be regenerated for use in the removal of the metal ions from the contaminated environment. Let us come to our conclusion that we have broadly isolated and characterized, and 16S rRNA showed that these two isolates were Bacillus cereus and Brevibacillus brevis. We are giving a message to the society that our investigation reveals that our organisms not only degrades chromium, but also it accumulates chromium. Thus, it changes it from the cyclical pool to the reservoir pool. Our organisms are potent leather degraders, thus they can be used in various leather industries for recycling and processing of leather. Our isolates have tremendous industrial application that it produces different types of industrial enzymes like the protease, like the keratinase, like the lipase. And this is the
Okay, let us
biosorption
carry on.
efficiency result. Hello?
Yes. Okay.
So
Thank
the
you, Mr.
just one line that this 200 μg per ml at the optimized condition, and we are giving a message to the society that this bioremediation and bioabsorption can be a cost-effective procedure if one organism can be used for tackling different environmental problems. Here, IR isolates appears to be multidimensional. They are excessive bioremediation ability, bioabsorption ability, as well as they are also industrial relevant. These are my reference, and I would like to thank all my respected scientists and professors, D.L.S. PG College Bilaspur, and the Microbiology Society India for organizing this international seminar for the advancement of science. Thank you, and thanks for patience.
Thank you very much, Madam. Okay, thank you so much. Next participant. कौन सा नंबर? कौन है?
Yes, yes, sir. Yes, madam. Ji, I am
Please
Balram.
turn off your video.
Yes, sir.
बलराम नंबर?
Yes, sir. Yes, sir.
बलराम को ले देगा नंबर?
Yes, sir. चालू कर दूं मैं?
Screen is visible. Please continue.
म्यूट हो गया ना? हो गया, हो गया. ओके, ओके. स्क्रीन शेयर करो ना. स्क्रीन शेयर.
बलराम को रहे बोल रहे? हाँ.
Balram, your audio is off. First turn it on.
Next participant, Neha Kausar.
Yes, I will share the
ये कौन है? ईना?
Ina Kausar.
Ma'am, please start.
ईना कौन से?
हो रही है मैम?
Yes, Ina, please continue. Slide is visible. Yes, sir. Yes.
Under the Seema Blorkar ma'am, Department of Microbiology, University, Bilaspur, and I'm here to present my piece of work. The title is Bioconversion of Fruit Peel, that is agro-residues biomass for high-yield xylanase production using mixture designing. So the main aim of this study is to evaluate fruit peel waste as a low-cost substrate for xylanase production and fermentation, and to identify the most effective supplement for maximize enzyme yield. So in India, waste is a big challenge. India generates approximately 15 million tons of fruit peel waste annually from processing industries. These peels are rich in lignocellulosic material, जिसका एंड में लैंडफिल किया जाता है, जो कि एनवायरमेंटल पॉल्यूशन और ग्रीन हाउस गैसेस के एमिशन में बहुत कंट्रीब्यूट करता है। और साथ ही जो इसका एक एडवांस 30 to 40% टू 25% हेमिसोलिल होता है, जो कि एज अ न्यूट्रिएंट होता है, एक एक्सीलेंट सब्सट्रेट होता है फॉर माइक्रोबियल एंजाइम प्रोडक्शन के लिए। तो इसको मेनली ट्रांसफॉर्म किया जा सकता है वेस्ट को इनटू वेल्थ में। बट यहाँ पर एक और दिक्कत है कि एंजाइम प्रोडक्शन में कुछ हर्डल्स हो सकते हैं। लाइक ट्रेडिशनली एंजाइम के प्रोडक्शन में पहले क्या होता था कि सिंथेटिक किया जाता था, जो कि बहुत एक्सपेंसिव होता है, और प्योर सबस्ट्रेट का यूज किया जाता है, जो कि इंडस्ट्रियल स्केल में करना इकोनॉमिकली बहुत बड़ा चैलेंज है। तो करेंटली ये एक लिमिटेशन फेस किया जा रहा है। तो इसको ओवरकम करने के लिए जो सर्च फॉर सस्टेनेबल कॉस्ट इफेक्टिव ऑल्टरनेटिव हैज डायरेक्टेड attention towards agro-industrial waste biomass as a potential fermentation substrate, offering dual benefits of waste management and enzyme production. These are some in understanding xylanase enzyme properties and its industrial applications. Xylanases are enzymes that break down xylan, a key plant cell wall component, into simpler sugar like xylose and xylo-oligosaccharides by cleaving beta-1,4 glycosidic bond. xylan जो होता है वो सेकेंड मोस्ट अबंडेंट पॉलीसेक्राइड होता है इन नेचर में। इसका ये तो बेसिकली एंजाइम का कैरेक्टरिस्टिक्स है। इसके बहुत सारे एप्लीकेशंस है, लाइक यूज इन पेपर एंड पल्प ब्लीचिंग, बायोफ्यूल प्रोडक्शन, फूड एंड बेवरेजेस प्रोसेसिंग, एनिमल फीड एनहेंसमेंट, टेक्सटाइल के लिए। और इसका एनवायरमेंट बेनिफिट ये है, वन ऑफ द एनवायरमेंट बेनिफिट इज ऑल्टरनेटिव टू हार्श केमिकल प्रोसेसिंग इन बेसिकली इन पेपर इंडस्ट्रीज। तो ये क्या करता है, जो पेपर में डी इंकिंग या ब्लीचिंग के लिए केमिकल का यूज किया जाता है बेसिकली क्लोरीन का, तो क्लोरीन के यूज को कम किया जा सकता है, क्लोरीन के कंजम्पशन को, जो कि हार्श केमिकल होता है। तो इसको हम रिप्लेस कर सकते हैं एंजाइम के थ्रू। These are the current methods that xylanase production and its limitations. जो
conclude
basically
please.
होता
Conclude
okay, sir.
please.
So this is the overview of my work. मैं डायरेक्ट इसमें ही आप
Okay.
एक्सप्लेन कर देती हूँ।
कंक्लूड कीजिए, कंक्लूजन मैम।
सर कंक्लूजन ये बेसिकली ये है कि जो जितने भी ऑर्गेनिजम्स जो मैंने सेलेक्ट किए थे वो ऑर्गेनिजम्स सॉरी जितने भी मैंने सबस्ट्रेट सेलेक्ट किए थे वो सबस्ट्रेट जो होते हैं, और जो ऑर्गेनिज्म मैंने चूज किया था वो ऑर्गेनिज्म जो होता है उस सबस्ट्रेट का यूज करके जो मेन मेरा एंजाइम है का प्रोडक्शन हाई करता है। तो सारे जो एग्रो रेसिड्यूज है, फ्रूट पील्स है, जो पोटेंशियल था सोर्स ऑफ xylanase एंजाइम के प्रोडक्शन के लिए, इसमें बेसिकली जो पोमोग्रेनेट पील है वो एज अ सप्लीमेंट मोलेशियस उसमें डाला गया, तो वो हाईएस्ट यील्ड दिया xylanase का, दैट इज 27.57। और साथ ही साथ इसको जब मैंने मिक्सचर डिजाइनिंग के लिए उसमें मैंने कुछ कॉम्बिनेशन में पील्स का यूज किया था। उसमें जो था ऑरेंज पील एंड पोमोग्रेनेट पील, जो कि हाईएस्ट एंजाइम का प्रोडक्शन दिया, 6 IU पर ml एंजाइम था। तो
हिना, आपका जो ये जो आपने एक्सपेरिमेंट किया इसका बेसिक सिग्निफिकेंस क्या है? क्या निकलता है इसका सिग्निफिकेंस?
बेसिकली ये है कि जो एग्रो वेस्ट है उसको हम यूज कर एंजाइम का प्रोडक्शन कर रहे हैं, और ये जो एंजाइम मतलब पर्टिकुलर xylanase का और xylanase के बहुत एप्लीकेशन है जो अलग अलग इंडस्ट्रीज में यूज किया जा सकता है।
ओके ओके, ठीक है, थैंक यू।
बोल सकते हैं।
ओके ओके,
मैम
ओके, नेक्स्ट
पार्टिसिपेंट।
सर मेरा स्क्रीन शो हो रहा है सर?
नहीं हो रहा है।
शो नहीं हो रहा है सर?
नहीं हो रहा है।
ओहो।
क्या बलराम साहब, कहाँ से बोल, कोरबा से बोल रहे हो?
नेक्स्ट पार्टिसिपेंट।
नेक्स्ट कौन है मैडम?
नेक्स्ट पार्टिसिपेंट। विनीत विनीत कुमार पटवा, सविता यादव, मोनिका। एनीवन?
Is my screen visible, ma'am?
Yes, ma'am. You can start. Please share your screen.
Good afternoon, sir, and good afternoon, ma'am. मेरा नाम
गुड
सविता
आफ्टरनून।
यादव है।
सविता यादव, एक सेकंड सविता, कौन से नंबर पे है मैडम?
जी,
32।
मैं रिसर्च स्कॉलर हूँ कलिंगा यूनिवर्सिटी रायपुर से।
जी
मेरा आज का
सविता,
टॉपिक है सर। जी
सविता आपको 5 मिनट मिलेगा, ठीक है? 2:05 में आपका टॉपिक खत्म हो जाएगा, स्टार्ट कीजिए फास्ट।
जी जी ठीक है सर। मेरा टॉपिक है सर कि स्कूल पाठ्यक्रम में पर्यावरण शिक्षा नीति 2020 के दिशा निर्देशों का प्रभाव। राष्ट्रीय शिक्षा नीति 2020 भारत की स्कूली शिक्षा में पर्यावरणीय साक्षरता को सुदृढ़ रूप में शामिल करने का एक महत्वपूर्ण अवसर प्रदान करती है। यह नीति रटने पर आधारित शिक्षण के स्थान पर अनुभव आधारित, कौशल आधारित और बहुविषयक शिक्षण पर बल देती है, जिससे विद्यार्थी अपने आसपास के पारिस्थितिकी तंत्र, जैव विविधता, जल और भूमि संसाधन तथा मानव और प्रकृति के संबंधों को
सविता, आपका
वास्तविक रूप में
प्रेजेंटेशन, आपका स्क्रीन शेयर करिए ना प्रेजेंटेशन का।
पीपीटी नहीं बना है मैम मेरा।
ओके,
जी
चलिए पढ़िए।
चलिए पढ़िए फिर।
जी इस नीति में पर्यावरण शिक्षा को सभी कक्षाओं में अनिवार्य रूप से जोड़े जाने की आवश्यकता दोहराई गई है, ताकि यह शिक्षण प्रक्रिया का स्वाभाविक और केंद्रीय भाग बन सके। सतत विकास हेतु शिक्षा पर आधारित यूनेस्को का दृष्टिकोण भी यही संकेत देता है कि शिक्षार्थियों को ऐसे ज्ञान, कौशल, मूल्य और व्यवहार विकसित करने चाहिए जो पारिस्थितिकी तंत्र की सुरक्षा, जलवायु से निपटे और साधनों विवेकपूर्ण उपयोग में सहायक हो। यह वैश्विक दृष्टि राष्ट्रीय शिक्षा नीति में निहित पर्यावरणीय उद्देश्यों को और मजबूत करती है, क्योंकि इसमें व्यवहार परिवर्तन, सामुदायिक सहभागिता और प्रकृति पर आधारित अधिगम पर विशेष जोर देते हैं। सर, इसका निष्कर्ष
सविता,
यही है कि
सविता
राष्ट्रीय
मैं
शिक्षा
आपसे,
नीति। जी
सविता आप मेरी बात सुनिए। हेलो।
जी सर। हाँ सर।
हाँ, आप उसको पढ़ रही हैं जो भी लिखी है उसको, तो इसमें नेक्स्ट
जी
से क्या बोलना चाहती है, एक लाइन में बोल के कंक्लूड कीजिए फटाफट।
जी सर, मैं यही बोल रही थी कि इसका निष्कर्ष यह है सर कि राष्ट्रीय शिक्षा नीति के तहत स्कूली बच्चों को रटने के बजाय विषय को समझने पर जोर दिया जाना चाहिए।
चलिए, थैंक यू, बहुत बढ़िया, ठीक है।
जी जी थैंक यू सर।
थैंक यू। नेक्स्ट कंटिन्यू।
जी
नेक्स्ट पार्टिसिपेंट। Please share your screen and start. रानी श्रीवास, प्रेरणा जायसवाल।
सर मैडम मोनिका राग से।
Okay, ma'am. Please share your screen and start the presentation.
क्वेश्चन नंबर 20। डॉक्टर मोनिका।
यस सर, गुड आफ्टरनून। हेलो।
Yes, ma'am, you are visible. Please continue.
यस Okay. Effect of xenobiotics on drug metabolism and drug-drug interaction. So first, xenobiotics. Effect of xenobiotics. First, xenobiotics. xenobiotics एक ऐसा सब्सटेंस है जो हम हमारी बॉडी में हमारे फूड के साथ, xenobiotics आर द सब्सटेंस दैट आर फॉरेन टू द लिविंग ऑर्गेनिजम्स, मतलब जो हमारी लिविंग बॉडी है उससे जो भी अदर देन चीजें हैं वो सारी xenobiotics में आती हैं, है ना। जैसे फॉर एग्जांपल हमारी ड्रग्स हो गई, हमारे फूड एडिटिव्स हो गए, मतलब फूड्स के साथ जो हम एडिशनल चीजें लेते हैं, खाते हैं, है ना। सिंथेटिक पॉलीमर्स हो गए, हमारे ऑयल मिक्सचर्स हो गए, पेस्टिसाइड्स हो गए, इन्वायरमेंट में जितने भी पॉल्यूटेंट्स हैं वो सारे, मतलब फ्रेश एयर को छोड़ के जितनी भी चीजें हैं वो सारी जीनोबायोटिक्स में आएगी। और ये हम कॉमनली क्या कर रहे हैं कि हमारे लाइफ स्टाइल ऐसी है कि हम लोग जो हमारी बायोलॉजिकल सिस्टम है उसके साथ इन सब चीजों को इनटेक करते हैं, है ना। जैसे कि हम डब्बा बंद चीजें खाते हैं, उसमें कुछ प्लास्टिक का कंटेंट जाता ही है, और भी चीजें जो सारी की सारी इन्वायरमेंटल पोल्यूटेंट्स में आती हैं। तो अब ये कहीं ना कहीं हमारी बॉडी के अंदर जा रही है और कहीं ना कहीं टॉक्सिक चीजें प्रोड्यूस कर रही है। तो इन सब चीजों को हमारी बॉडी से बाहर कैसे निकालना है, उसके लिए हमारी जो है कि रिएक्ट करती है कैसे, एक्ट करती है कैसे, इंटरेक्ट करती है, और इसका मेटाबॉलिज्म कैसे होगा बॉडी के अंदर। तो इस चीज पे मैंने थोड़ा सा स्टडी किया है, कोई रिसर्च नहीं किया हुआ है। तो उसके लिए मैंने थोड़ा सा ये देखा कि भाई जो हमारी बॉडी है वो ऐसी चीजें इंटेक करे जो कि बायो ट्रांसफॉर्मेबल हो। जैसे कि जो हमारा लिवर है वो उसको, जो हमारा फेज 1 रिएक्शन है और फेज 2 रिएक्शन है, उन दोनों के अंदर जाए और एक लिपोफिलिक सब्सटेंस बन के, वाटर वाटर सॉल्युबल सब्सटेंस बन के, थ्रू यूरिन, थ्रू स्टूल्स बॉडी से बाहर हो सके। तो ऐसी चीजें जैसे कि हमारा फेज 1 रिएक्शन है, फेज 1 रिएक्शन में मेनली साइटोक्रोम P450 जो मेन रोल प्ले करता है कि फूड डाइजेशन में, उसके हाइड्रोलिसिस में, ऑक्सीडेशन में, रिडक्शन में, इन सारी रिएक्शंस में फेज 1 रिएक्शन जो होती है वो प्ले करता है। और जो फेज 2 होता है जिसमें मेनली कॉन्जुगेशन रिएक्शंस होती हैं ग्लूकोरोनिक एसिड की और सल्फेट का एडिशन होता है, और उसके बाद वो पोलर मॉलिक्यूल्स में कन्वर्ट होती है। तो इसलिए हम ऐसा सोच रहे हैं कि भाई हम हमारा जो भी एडिशनल सब्सटेंसेस लेते हैं वो हो सके तो ऐसा लें कि उसमें टॉक्सिसिटी कम प्रोड्यूस हो। दे कैन बी टॉक्सिक एंड कॉज म्यूटेशन, जो हमारे हॉर्मोन्स में जो म्यूटेशन आ रहा है आजकल आप देखेंगे कि सडन अटैक आ जा रहा है, सडन बहुत सारी चीजें सडनली हो जा रही हैं। तो उसके कहीं ना कहीं रीजंस जो हैं, जो हमारी नॉर्मल लाइफ है उसमें हम लोग ऐसी क्या चीजें कर रहे हैं उसको मिनिमाइज करने की कोशिश करना, और ऐसी चीजें यूज करना कि अगर वो जा भी रही हैं तो वो मेटाबोलाइज प्रॉपरली हो जाए। तो इसके लिए फेज 1 में बायो एक्टिव सब्सटेंसेस में P450 साइटोक्रोम वाला है, और दूसरा ग्लूकोरोनिक एसिड एस ट्रांसफरेजेस का हम रोल देखते हैं, जिसमें जो फंक्शनल रिएक्शंस होती हैं जैसे कि अभी मैंने बताया कि लिवर मेन रोल प्ले करता है, इसके साथ साथ जो इंटेस्टाइन है, किडनी है, लंग्स हैं, और स्किन, ये भी कुछ ना कुछ परसेंट में रोल प्ले करती है। जैसे कि हमारी स्किन है, स्किन से स्वेट निकलता है, तो जो स्वेट है कहीं ना कहीं उसमें भी इम्प्यूरिटीज आती हैं। जो हमारी लंग्स हैं, जो हमारी कुछ परसेंट में एयर के साथ एक्सेल आउट होती है। देन किडनी है, उसके में भी हमारे पॉइजनस सब्सटेंसेस काफी हद तक बॉडी से एक्सक्रीट आउट हो जाते हैं। तो
मैडम
इसलिए,
प्लीज कंक्लूड कीजिए।
ओके कंक्लूड। तो जो मैं कहना चाह रही थी कि भाई जो बॉडी में ड्रग का इंटरेक्शन है, जैसे कि मान के चलिए हमारे जो भी ड्रग का इफेक्ट है, जैसे कि हमारे स्मोकिंग हो गई, अल्कोहल हो गया, रिफेम्पिसिन हो गया, तो जो जो ड्रग्स हैं वो हमारी बॉडी के अंदर जाके या तो इंडक्शन करते हैं या इनिबिशन करते हैं, या हमारी बॉडी के नॉर्मल सब्सटेंसेस के साथ कंपटीशन करते हैं। और कंपटीशन ऐसा होना चाहिए कि हमारा जो नॉर्मल बॉडी फंक्शन है वो जीत जाए कंपेयर टू ड्रग। ऐसा कुछ हम लोग सोचते हैं कि भाई ऐसा कुछ हो
चलिए
कि भाई ये चीजें
चलिए,
आ जाए।
थैंक यू मैडम, थैंक यू।
ओके, Thank you so much, sir.
Thank you, madam. Next, please.
Next participants, please be ready and present your PPT here. प्रेरणा जायसवाल, प्राची, प्रियंका साहू, रानी श्रीवास, रितंभरा चौहान, सरस्वती, सुशील, is there anyone available for presentation? Whosoever is ready with the PPT, please share your screen and start the presentation.
कल से
बलराम सर।
जी सर।
चलिए जल्दी प्रेजेंट कीजिए सर।
इसी हो रहा है सर, सॉरी सर, मैं क्या बताऊं।
नहीं, कोई दिक्कत नहीं, जल्दी करिए।
जी सर, ऐसे बोल रहा हूँ सर मैं। जी सर। सर मेरा टॉपिक था सर, स्टडी ऑफ स्पेशियो टेम्पोरल पैटर्न ऑफ फिश डाइवर्सिटी अक्रॉस 4 मेजर रिवर ऑफ डिस्ट्रिक्ट कोरबा, छत्तीसगढ़। सर मैंने सर ये 4 रिवर को यहाँ पर स्टडी के लिए लिया था सर।
वीडियो ऑन कर लीजिए सर, खाली नाम दिख रहा है आपका, वीडियो ऑन कर लीजिए।
वीडियो ऑन कर, यस सर मैं बोल रहा हूँ सर। यस सर,
प्लीज कंटिन्यू।
जी सर, जी सर। तो सर मेरा स्टडी पीरियड 6 मंथ का था सर, और इसमें मैंने 4 रिवर को लिया है कोरबा जिला में सर। हसदेव रिवर, चोरनई, तान एंड अहिरन रिवर सर। इसका सर 6 मंथ का स्टडी के दौरान मैंने सर ऑब्जर्व किया कि इसमें लगभग अलग अलग नदियों में अलग अलग प्रकार के फिश डाइवर्सिटी देखने को मिल रहा है सर। हसदेव नदी चूंकि मेन लाइफ लाइन ऑफ कोरबा है, वो उसमें फिश डाइवर्सिटी अधिक देखने को मिल रहा है सर। इसमें मैंने अपने स्टडी के दौरान 4 स्पीसीज जो रिकॉर्ड किया है सर, वो अभी फर्स्ट टाइम ऑब्जर्व किया है, जिसमें से है सर मोनोप्टेरस कुचिया करके है, और एंगुला बेंगालेंसिस, और गर्रा मुलिया, और एक और न्यू स्पीसीज रिपोर्ट किया है चेला कैचियस। ये 4 स्पीसीज मैंने सर न्यू रिपोर्टेड है। महानदी में मिलता था ये, लेकिन ये फर्स्ट टाइम ये हस्ते रिवर मैंने ऑब्जर्व किया है सर। तो सर मैं कंक्लूजन में आता हूँ सर, चारों रिवर को यदि हम कंपैरेटिव स्टडी मैं किया हूँ सर, तो चूंकि हसदेव नदी लंबाई और प्रवाह ये काफी बड़ा होता है, और इसमें मैंने स्टडी एरिया में मैंने अप स्ट्रीम, मिड स्ट्रीम एंड डाउन स्ट्रीम को लिया था सर, और साथ ही मैंने प्री मानसून, मानसून और पोस्ट मानसून, ये तीनों सीजन को कवर किया था सर। तो मेरा अध्ययन में मुख्य ये था सर कि समय और स्थान के आधार पर मछलियों की विविधता में क्या अंतर आता है। तो मैंने अपने स्टडी में पाया सर कि जो मानसून सीजन है जनरली, इसमें फिश जो है अपना हाईएस्ट डाइवर्सिटी शो करते हैं सर, और साथ ही जो पोस्ट मानसून है, चूंकि इस समय जो वाटर है जो स्टेबल कंडीशन में आ जाता है, तो मानसून एंड पोस्ट मानसून है ना, दैट 2 सीजन शोज हाईएस्ट डाइवर्सिटी, बट प्री मानसून सीजन शोज लोवेस्ट डाइवर्सिटी। इसी प्रकार सर यदि हम स्थान को देखें सर, तो जो रिवर का अप स्ट्रीम रीजन है, अप स्ट्रीम रीजन जो है, क्योंकि वहां पर नेरो चैनल होता है और साथ ही फास्टर मूव होता है वाटर का, तो वहां पर स्पीसीज डाइवर्सिटी कम देखने को मिलता है सर। और साथ ही मिड स्ट्रीम जो होता है, जो हेड वाटर बॉडी होता है सर, वहां पर फिश का डेंसिटी एबंडेंस अत्यधिक देखने को मिलता है सर, यानी कि यहाँ पर हाई डाइवर्सिटी शो होता है सर। और साथ ही साथ जो डाउन स्ट्रीम होता है सर, क्योंकि डाउन स्ट्रीम यदि हम हसदो की बात करें साहब, तो यहाँ पर संगम होता है हिरन और तार रिवर का, तो यहाँ पर जो संगम स्थल है वहां पर अत्यधिक मिलता है सर। मैं सर इसमें कंपैरेटिव स्टडी भी बनाया सर चार्ट के माध्यम से, सॉरी मेरा
कंक्लूड कर दीजिए,
नहीं हो पाया। तो सर मैं अध्ययन में निष्कर्ष ये निकलता है सर कि हमारे जो फिश की विविधता है, इसमें स्थान और समय का बहुत महत्व है सर। अलग अलग स्ट्रीम में अलग अलग समय पर मछलियां अपने सीजन के दौरान अपनी विविधता और उनकी संख्या में परिवर्तन आता है सर। सॉरी सर।
थैंक यू सर। और सब बढ़िया है सर?
सब ठीक है सर, आइए, वेलकम पटेल सर। जी सर, ठीक सर, नमस्कार सर, नमस्कार सर, थैंक यू सर।
Next participant, Please present your PPT here and start the presentation.
हाँ हेलो, नमस्ते मैम।
नमस्ते, क्या नाम है भैया आपका?
डोला मैंने साहू बोल रहा हूँ।
अच्छा, कौन सा नंबर का है? 2 नंबर,
15 नंबर।
15 नंबर पे। कहाँ से है डोलामणि जी?
जी कोरबा गवर्नमेंट पीजी कॉलेज कोरबा से।
कोरबा से, चलिए ठीक है। पीपीटी शो क्या करेंगे?
सर वही दिक्कत हो रहा है थोड़ा हम लोगों से अभी, मैं
अच्छा
इसमें बोल देता हूँ सर।
अच्छा, चलिए ठीक है, बोलिए। आपका 2:20 पे खत्म हो जाएगा टाइम, फटाफट बोलिए।
जी जी सर, मेरा जो टॉपिक है वो एक रिव्यू सर्वे है, सोशियो इकोनॉमिक इम्पोर्टेंस ऑफ मशरूम और कोरबा डिस्ट्रिक्ट से रिलेटेड है।
ठीक है, बोलिए।
मशरूम एक बहुत ही मेडिसिनल और न्यूट्रिशनल फूड है, एक नॉन टिम्बर फॉरेस्ट प्रोडक्ट के रूप में है, जो ग्रामीण क्षेत्र के लोगों के द्वारा इसको सेवन किया जाता है, मार्केट में इसको बेचा जाता है। इससे जो उनको आर्थिक सहयोग होता है उनके जो फैमिली इनकम में, इससे रिलेटेड सर्वे था। इसमें सामान्य रूप से कोरबा चूंकि एक ट्राइबल डिस्ट्रिक्ट है, इसमें गोंड, उरांव और गंवार जो जनजाति के लोग हैं, ये ज्यादातर अपने लाइवलीहुड के लिए मशरूम का, वाइल्ड मशरूम का उपयोग करते हैं। इस रिव्यू का जो एम था सर कि मशरूम के जो न्यूट्रिशनल, मेडिसिनल, इकोनॉमिक वैल्यू के बारे में जानना था, और ग्रामीणों के बीच उनके जो यूज हैं, फूड के रूप में हो चाहे मेडिसिनल के रूप में, इसके लिए जानना था। उनके पास जो चैलेंजेस आते हैं कलेक्शन के दौरान, मार्केटिंग के लिए, उन सब से रिलेटेड था। मशरूम चूंकि बहुत
इसको
ही
कैसे कंक्लूड करेंगे प्रोडक्शन के बाद? इसका मेन ये क्या है आपका कंक्लूजन पॉइंट, क्या है बताइए ना एक चीज, एक लाइन में
कंक्लूजन ये सर कि जो मशरूम, वाइल्ड मशरूम का ग्रामीण लोगों के आजीविका में ठीक ठाक योगदान है। 10 से 12% की वो आजीविका में उनका योगदान है वाइल्ड मशरूम
ठीक है
का।
ना, इकोनॉमिक इम्पोर्टेंस समझ गया। और सेकंड और कुछ क्या बताना क्या चाह रहे हैं इसमें?
इसमें इसमें जो प्रोडक्शन हो रहा है, जो
प्रोडक्शन भी समझ गए। और क्या बताना चाह रहे हैं? आपका
वो
सब्जेक्ट
मेन
क्या है
पर्पस था
डोलामणि
सर,
जी?
वाइल्ड मशरूम और कल्टीवेटेड मशरूम का इम्पैक्ट।
अच्छा, आपका सब्जेक्ट क्या है? बॉटनी है, जूलॉजी?
मेरा बॉटनी है सर।
बॉटनी है, ठीक है। इसका इसका कुछ रिसर्च बताइए ना हमको। क्या इकोनॉमिक इम्पोर्टेंस, कल्टीवेशन, प्रोडक्शन से कोई मतलब नहीं।
सर ये मेरा एक्चुअल में रिव्यू आर्टिकल था, जो पुराने
अच्छा,
लिटरेचर है उससे
ठीक है,
रिलेटेड मैं।
चलिए, थैंक यू, ठीक है, कंटिन्यू करते
थैंक
हैं। ओके, थैंक यू वेरी मच, थैंक
Very much. Next participants, please. प्राची, प्रेरणा जायसवाल, प्रियंका साहू, दानिश श्रीवास, रितमजा,
यू। कौन आ रहा है?
अनुपमा, I think want to present.
सबका लंच टाइम हो गया? ये अनुपमा आरती का हो तो दूसरे। हाँ, बोलिए।
Anyone wants to present?
चलिए, जो भी इंटरेस्टेड है फटाफट आइए, प्रेजेंट कीजिए।
वंदिता जेना,
कौन है? कौन है? अरे एक घंटे में सब रही गई। जल्दी तो नहीं करना चाहिए।
If anyone interested to present, please switch on your mic and start presentation. Please share your screen and start the presentation.
Hello, ma'am. I'm Anupama. I have a presentation. Can I do it now?
Yes, yes, yes.
I'm from
Yes,
Chennai.
yes, ma'am. Please show.
My presentation is about the analysis of textile dye effluents.
Ma'am, please share our PPT or turn on your video. Anupama ma'am, please turn on your video. If you
The
have
system is showing some problem. I have a connectivity issues from my end. So I'm just calling from. Is it okay?
You can switch on your video, ma'am.
Video, in the sense I'm having system issues with my personal system.
Okay,
So,
okay, ma'am. Start, ma'am, please.
video in the sense my pardon. I just
Okay,
come
please.
outside and.
Okay, ma'am. No problem. Please start.
So I just done bioremediation using my, because I can hear your voice partially only due to some issue network connectivity. Shall I-
In the meanwhile, if anyone else is having the presentation, please share your screen and start the presentation if anyone is available.
to the environment. So this particular textile dyes are usually toxic, carcinogenic, and persistent in the environment. Am I audible, ma'am?
Your voice is breaking, ma'am.
Because it's breaking, small sort of issue.
No problem, ma'am. Please start.
Bioremediation of textile dyes is a promising approach to mitigate the environmental impact of textile industry. It is a cost-effective method and eco-friendly alternative to conventional treatments. So I've chosen this methodology to inhibit or to reduce the use of chemical dyes in the particular thing has to be totally decreased when it is let into the environment. In other words, to mitigate the impact. The objectives were to collect this effluent samples from textile units and to isolate the potential degrading bacteria and to evaluate the efficiency of the bacterial strains in various dyes using a prominent technology. So the process or the methodology was like this. So first I collected the textile effluents from Tirupur. That is a main area where in Tamil Nadu, the particular textile dyes or textile units are being there. These samples are transported in sterile containers at 4 degrees C. It was filtered using Wattman number filter paper. Then each 5 ml of textile dye sterile effluent was inoculated with the bacterial inoculum, which we were able to isolate in minimum salt media. So 5% of the bacterium inoculum was added. One control test tube without bacteria was also simultaneously used to assess the natural degradation. This was done consecutively for 7 days by incubation at 37 degrees centigrade in a shaker incubator. The 24-hour intervals were chosen for analysis and the absorbance was measured using the particular UV visible spectrophotometer. Finally, the particular results were tabulated and we calculated the decolorization percentage by using this formula A0 divided by A0 by AT into 100, where the initial absorbance, that is before bacterial inoculation was taken into account, then the absorbance of dye solution after incubation was subtracted from this, and this was in the denominator, and we multiplied with 100 to get the percentage. we got several isolates, of which 5 isolates were finally utilized for this particular activity, showed up good results. 99.8% was the highest one shown by one of our isolate, and thus was chosen to be the best isolate. It is subjected to phylogenetic analysis. I just
Madam,
give,
please conclude.
going for, pardon me? Sir?
Madam, please conclude.
Ah, okay.
Conclude, madam.
So one particular isolate was being good enough for further evaluation. Thank you, sir.
Thank you, madam. Thank you. मैडम नेक्स्ट पार्टिसिपेंट ले लेंगे.
Hello.
If anyone is available, please share the screen and start the presentation.
Good afternoon, ma'am. I'm Ritambara Chauhan, Assistant Professor Biotechnology, GNAPG College. Can I start my presentation?
Yes.
Can I do it right now?
Yes.
Ritambara?
Yes, sir.
Yes, ma'am. Pitambara?
Yes, ma'am. Yes,
Your name, please,
ma'am.
ma'am.
Ritambara Chauhan.
Pitambara Chauhan.
Okay, sir. So, sir, my topic is xenobiotic pollutants. Xenobiotic pollutants are generally the pollutants which are foreign for living organisms. All those pollutants are grouped in this category of xenobiotic pollutants. These are chemical substances. Generally, these are recalcitrant or synthetic molecules which are present in our environment and in our body. Their concentration is unnaturally very high, and these compounds like chemical fertilizers, pesticides, and dyes, and additives like some chemicals like polychlorinated biphenyls and polycyclic aromatic hydrocarbons. These are some examples of xenobiotic pollutants. The main sources of these pollutants come from agriculture and industrial chemicals, environmental pollutants also, and our lifestyle and diet, like the additives present in our food. These are all the sources. Some natural toxins which are found in plants and fungi, algae are also come in this category of xenobiotic pollutants. Pharmaceutical and personal care products are also the sources, and some water and food contaminants
488541
are the main sources of this. Some heavy metals also include the xenobiotic pollutants, and their impact is very serious on environment and health. Like in human beings, these xenobiotic pollutants cause various type of diseases like neurodegenerative diseases and heart problem, cardiac problem also. And if long time exposure of these xenobiotic compounds are faced by human beings, then there are also the chances of cancer also. So their long-term exposure is very harmful. Apart from these, plant and water also are polluted by these xenobiotic compounds, and in plants this compound affect their electron transport chain, and various morphological and physiological disorder are also seen in plants by these compounds. And the soil and water are very much contaminated by the industrial effluents of xenobiotics. Now I come to the biodegradation of xenobiotics. So this degradation of these xenobiotics is very crucial, because it's causing very much it's causing our environment very much polluted, and the process of breakdown of this compound by microorganism has been studied by many scientists. And this process in which microorganisms degrade these xenobiotic compounds is known as bioremediation, in which we use microorganisms which are naturally found in the environment and degrades these toxic substances. Bacteria and fungi have been identified because they have some unique properties that enable them to partially or completely metabolize the xenobiotic substances. And also these microorganisms are affected by various factors like temperature and their nutrition sources and pH. All these factors, physical factors, which affect their growth and availability, affect their degradation process. Now, mechanism of biodegradation. We know mainly two main pathways are adopted by the microorganisms to degrade these xenobiotic compounds. These are aerobic biodegradation and anaerobic biodegradation. As we know, in aerobic biodegradation, the presence of oxygen, microorganisms use enzymes to degrade the wide range of compounds, and mainly the benzene, phenol, toluene, hydrocarbons like aliphatic compounds. These are degraded by aerobic bacteria in the presence of oxygen. And the key enzymatic reactions of this aerobic biodegradation are oxidations catalyzed by oxygenase enzyme and peroxidase enzymes. Some example of aerobic degradative bacteria are Pseudomonas and Bacillus species. Apart Micrococcus species have also been identified to degrade xenobiotic compounds. The next is anaerobic biodegradation. In anaerobic biodegradation, the synthetic and naturally found xenobiotic compounds are degraded by microorganisms in environment lacking oxygen. This process is mainly useful for polychlorinated biphenyls, dioxins, and some pesticides like DDT, which is used in very much amount by the farmers. And examples of anaerobic microbes include D.Sulfovibrio species, Methanospirillum species, and methanobacteria are mainly in these categories of anaerobic biodegrading microorganisms. Yes, sir.
Please conclude karo.
Yes, sir. Yes, sir. Yes, sir. Okay. So last two slides only left, sir. I have only mentioned here the biodegradation of petroleum products, and petroleum products are biodegraded by mainly two aspects or two methods known as bioaugmentation and biostimulation. In bioaugmentation, we introduce the endogenous microorganism in the environment, and they are capable of biodegrading these xenobiotic compounds in their natural environment. And in biostimulation, we use microbes are there present, but they are lacking some important nutrients, because of which they.
Thank you.
Okay, sir. Okay, thank you, sir.
Thank you.
Next student. Next any participants, please share your screen and start the presentation. If anyone is present here, please.
Hello, this is Mitesh Nema. Can I present, madam?
Yes, please.
Sir, this is Mitesh Kumar Nema.
Mitesh Kumar Nema, 39. Please share
Okay, ma'am. Okay.
Please share your screen and start the presentation.
Is it visible, ma'am? Is it visible?
Yes, visible.
Ji. Good afternoon, everyone. I am Mitesh Nema, Research Scholar, Department of Botany from Dr. C.V. Raman University, Kargi Road, Kota. And now I am here to represent my topic, and that is fungal frontiers in nutritious grains, exploring microflora diversity in open market millets for safer food system. This is overview that I have discussed later. फर्स्ट ऑफ ऑल हम सभी जानते हैं कि मिलेट्स को हमारी इंडियन गवर्नमेंट ने 2018 में न्यूट्रिशियस ग्रेन्स के रूप में कहा गया है, और अभी वर्तमान समय में मिलेट्स काफी ज्यादा मात्रा में पौष्टिक ग्रेन के रूप में काफी प्रमोट किया जा रहा है, जिसमें हम मुख्यतः रागी, कोदो, मिलेट, बाजरा को काफी बहुतायत में यूज करते हैं। तो मेरी ये जो रिसर्च है ये मुख्यतः इन मिलेट्स में पाए जाने वाले अनहाइजीनिक हैंडलिंग और उनके जो पुअर स्टोरेज और हाई ह्यूमिडिटी के कारण होने वाले माइक्रोफ्लोरास हैं, फंगस ग्रो उसमें करती है, उनके कंटामिनेशन पर है कि इस वर्क में मैं उन फंगस को आइडेंटिफाई करूंगा कि किस आधार पर ये फंगस जो है मिलेट्स पर अपना इफेक्ट करती है। तो मुख्यतः ओपन मार्केट्स जो बिलासपुर के डिफरेंट मार्केट्स हैं उनमें पाए जाने वाले मिलेट्स के ऊपर इस काम को किया जाना है। और इसमें हम द रेंज ऑफ जो फंजाई है मिलेट सैंपल्स पर उन पर काम करेंगे और इन फंजाई को किस तरह से हम इम्प्रूव करके इम्प्रूव ड्राइंग एंड स्टोरेज कंडीशन को हैंडलिंग करके मैक्सिमम तरीके से फंजाई को ग्रो होने से रोक सकते हैं, उस पर काम करना है। जिसमें इसमें रिव्यू ऑफ लिटरेचर है, ये कुछ रिसर्च और रिव्यूज हैं जिन्होंने मुझे अपने रिसर्च को करने के लिए काफी हेल्प किया हुआ है। और इसकी इम्पोर्टेंस में देखा जाए तो द अंडरस्टैंडिंग द फंगल डाइवर्सिटी एसोसिएटेड विथ मिलेट्स इन एसेंशियल फॉर इंश्योरिंग food security and public health, because these nutritious and affordable super grains are very highly valuable in fungal infestation during market display, ट्रांसपोर्टेशन एंड स्टोरेज कंडीशंस पर काफी मात्रा में इनमें फंगस ग्रो कर जाते हैं। और मुख्यतः जो फंगस ग्रो हो रही है वो एस्परजिलस पेनिसिलियम फ्यूजेरियम जेनरा के फंगस ग्रो करते हैं, जो कि उसके फूड क्वालिटी में कार्बोहाइड्रेट प्रोटीन वसा की क्वालिटी पर काफी इफेक्ट डालते हैं, जो कि मिलेट्स के ग्रेन्स को कई तरह से प्रभावित करती है। तो इसमें इट आल्सो सपोर्ट्स दिस रिसर्च वर्क फॉर पॉलिसी मेकर्स to establish safety standards, help research identify contamination patterns and research resistant millet varieties, कि हम कैसे मिलेट वैरायटी को रेजिस्टेंट बना सकते हैं, कैसे हम कुछ ऐसी पॉलिसीज बना सकते हैं जो कि फंगस को ग्रो करने से रोके और फार्मर्स के लिए और हमारे शॉपकीपर्स के लिए स्टोरेज कंडीशन को बेहतर तरीके से बना सकें। तो दिस माय वर्क इज mainly based on traditional storage practices and handling methods and environmental conditions influences fungal proliferation and potential mycotoxin problems. So this study implies culture dependent microscopic techniques then that identify, isolate and characterize contamination fungal species. These findings contribute to bridging agricultural microbiology, public health, and food safety by highlighting fungal risk in open market millets and offering a practical solution to ensure safer and more sustainable food system. Some objectives of this research work is to use morphological and cultural traits to separate and identify fungal species linked to stored millet samples, and that stored millets are bajra, kodo, and ragi from open markets, and to use quantitative and visual techniques to document and assess the colony count, frequency, and abundance to isolate fungi species, and then to evaluate the effects of handling and storage by compare.
आप कंक्लूड करिएगा ना अपने रिसर्च को।
जी मैम, by comparing the biochemical deterioration, and then last one is to baseline data on millets mycoflora variety. So these are some objectives, and these is proposed methodology that sample collection, sample preparation, fungal isolation, purification of isolates. So these are some proposed methodology जिस पर वर्क करके मैंने ये प्राप्त किया है कि बाजरा के जो हमारा बाजरा मिलेट्स है उस पर फंगस ग्रो काफी ज्यादा मात्रा में आया हुआ है, और इसमें सबसे ज्यादा फंगस जो हुआ है वो एस्परजिलस फंगस ने अटैक किया था, जिसके कारण हम कुछ ऐसी पॉलिसी बना सकते हैं कि हम प्रॉपर हैंडलिंग करें और मिलेट्स को फंगल इन्फेक्शन से बचा सकें। तो इसमें मैंने जो प्राप्त किया उसमें बाजरा सबसे ज्यादा फंगल इफेक्ट हुआ, देन कोदो एंड देन रागी, एंड सबसे ज्यादा फंगस जो इफेक्ट था वो एस्परजिलस था, फ्यूजेरियम था, एंड देन पेनिसिलियम था। इसके अलावा साथ में प्रोटीन की क्वांटिटी लगभग 10% लॉस हुई, कार्बोहाइड्रेट की 6% एंड लिपिड की 5% हुई थी, ये भी एस्टिमेशन करके हमने पता किया था, एंड दैट इज।
सुशील कुमार पटेल, शैलनी फूल, सरस्वती साबिर खान, राजी
मैम हेलो Yes, sir.
Yes, Sabir, share your PPT.
ओके ओके सर स्क्रीन शेयरिंग नहीं हो पा रहा है प्लीज मैं
Dolamadi ji,
अपना
please wait for a while.
जी जी सर वो किन्हीं का चल रहा है
साबिर का नाम तो
नहीं है कौन सा नंबर हाँ
सर मैं आइडल हेलो हेलो
Yes, Sabir, you are audible and your screen is visible. Continue with your presentation.
Okay, sir. Thank you, sir. Very good afternoon to chairpersons and all the members here. I'm Shabir Khan. I am here to present my paper on the title of Isolation and Identification of Cryptophilic Fungi for Hair Dumping Yards and Poultry Soils. Now, these are my contents. And now, first is introduction. Keratinophilic fungi, it is a soil-dwelling fungi which can grow on soils. When it use keratin as a substrate to grow, and when keratinophilic fungi meet to the keratinous waste like hair, nails, feathers, etc., they produce an enzyme that is keratinase enzyme. That EC number of keratinase enzyme is 3.4.9911. Next is objective. What is the objective of this paper?
कंटिन्यू साबिर फास्ट
Hello, sir.
फास्ट करिए
Please raise
वेट अ सेकंड सर एक कॉल आ रहा है बस उसको हटा रहा हूँ हेलो सॉरी सर
कंटिन्यू फास्ट
first objective is isolation and second is identification, third is compare to the environments, and next is significance.
सिग्निफिकेंस क्या बताइए फटाफट
जी सर
सिग्निफिकेंस बताइए
सर significance is it is a public health concerns and biotechnological potentials. Sir, keratinophilic can produce an enzyme and they can be used in the biotechnological industries like biofertilizers and textile industries and pharmaceutical industries, bioplastic, etc. And they also can cause disease in some animals or humans. And okay, sir, biotechnological potentials is more important.
कंक्लूड कीजिए कंक्लूड कीजिए
Sir, I have isolated more than 8 genera of fungi and in Cryptosporiums, Mycosporium, Phycophytons, Fusarium, Aspergillus, etc. 140 total 142 isolates have occurred and 34% are pathogenic species.
ठीक है साबिर थैंक यू
Thank you, sir. These are my references.
Thank you very much. Next candidate, please. Please share your screen and start the presentation. Whosoever is ready with the PPT and presentation. Bandita Jena. Who has joined with the name Botany. Sushil Kumar Patel.
जी नमस्कार मैम मैं क्योंकि स्क्रीन शेयरिंग में दिक्कत हो रही है, मैं अपने टॉपिक का नाम बोल के अपने रिसर्च पेपर पढ़ना चाहूंगा, क्योंकि आप ध्यान दें।
Sir, your name, please.
Variation in composition, antioxidant activity, and microbial. We discuss silences, leaf and extract at Purba, Chhattisgarh. Silences,
अग्रवाल साहब आपकी वॉइस ब्रेक हो रही है।
शेयर करके नहीं बनी। हाँ
नेटवर्क में
सर, फ्लोरा ब्रिटिश इंडिया सर, कॉलेज का वाईफाई से चल रहा है सर। हाँ,
थोड़ा सा
फ्लोरा
दरवाजा
ब्रिटिश
खोलिए
इंडिया से
आवाज।
हमने आइडेंटिफाई किया। जी सर। सर, इसकी फाइटोकेमिकल कंपोजिशन का सीजनल वेरिएशन पे स्टडी किया है हमने। साल में 3 सैंपलिंग करके इसकी क्वालिटेटिव कैरेक्टर्स का स्टडी किया है, जिसमें हमने केवल लिग्निन कंपोजिशन पे वेरिएशन पाया है। इसके पश्चात इसकी क्वालिटेटिव कैरेक्टर्स का अध्ययन किया गया, जिसमें फ्लावर और लीफ के एक्सट्रैक्ट का टोटल एंटीऑक्सीडेंट एक्टिविटी देखा गया है। हिबिस्कस रोजा की लीफ की अल्कोहलिक एक्सट्रैक्ट से 40% टोटल एंटीऑक्सीडेंट आइडेंटिफाई किया गया है। वही फ्लावर से हमको 50% टोटल एंटीऑक्सीडेंट एक्टिविटी का प्राप्ति हुआ है, जबकि इसकी जो लीफ की वाटर एक्सट्रैक्ट से हमको 32% एंटीऑक्सीडेंट एक्टिविटी प्राप्त हुई है। सर, इसकी जो टोटल फिनॉल कंटेंट्स है, तो हमने लीफ का अल्कोहलिक एक्सट्रैक्ट लिया है, जिसमें 0.44 मिलीग्राम प्रति पर लीटर के अकॉर्डिंग इसकी टोटल फिनोलिक कंटेंट टीपीसी प्राप्त हुई है। वहीं पर हिबिस्कस की फ्लावर का अल्कोहलिक एक्सट्रैक्ट से 0.67 मिलीग्राम प्राप्त हुआ है, जबकि हिबिस्कस की लीफ वाटर में टोटल फिनोलिक कंटेंट प्राप्त हुआ है। इसका जो
आपकी आवाज बहुत ब्रेक हो रही है, बहुत ज्यादा ब्रेक हो रही है, ऑडिबल नहीं है, आपकी आवाज बहुत ज्यादा ब्रेक हो रही है। कंक्लूड कीजिए जरा।
सर, इसकी आपने माइक्रोबियल एक्टिविटी का अध्ययन किया है। इसकी माइक्रोबियल एक्टिविटी हमने कुछ एस्चेरेजिया कोलाई, स्टेफाइलोकाकस ऑरियंस।
So, can we take the next participants, please?
मैं अपना वीडियो बंद करता हूँ, शायद वीडियो से ठीक हो जाए। हाँ, अब बताएंगे। हाँ, एस्परजिलस नाइगर, स्टेफाइलोकाकस एपिडर्मिस, ये 4 माइक्रोब्स पे कार्य किया गया है, और हमने किया है तो हमने पाया कि जो मैक्सिमम एक्टिविटी है वो हमको स्टेफाइलोकाकस ऑरियंस में फ्लावर एक्सट्रैक्ट से प्राप्त होता है।
ठीक है अग्रवाल साहब, थैंक यू। चलिए, बहुत डिस्टर्बेंस है।
प्लीज दीप्ति गुप्ता।
मैम, हाँ, फिर से कंटिन्यू करूं क्या मैम? एग्जिट हो गया था।
नहीं, चलिए कोई बात नहीं,
कंटिन्यू सर।
ठीक है, रहने दीजिए अग्रवाल साहब।
नेक्स्ट, हु जॉइंड विद द नेम बॉटनी?
मैम, इसमें कंटिन्यू करूं?
Next participant, आरती साहू, बंदिता जेना। आरती साहू has started.
Bharati Sahu, if you are not able to screen your share, please turn on your video and do presentation.
Ma'am, please turn on your video and start the presentation.
गुड आफ्टरनून मैम एंड सर. सॉरी मैम, स्क्रीन शेयरिंग नहीं हो पा रहा है मेरे से.
No problem, ma'am. Please start your presentation.
Myself Aarti Sahu ma'am, Department of Life Science Botany, University, Dr. C.V. Raman University, Kargi Road, Kota, Bilaspur. My topic: integrative approach for eco-friendly management of lantana camera, role of herbicides, microbes, and phytoremediation strategies. So, lantana camera is one of the most weed spread and aggressive invasive weed found across tropical and subtropical regions, including large part of India. It adapts quickly to different climate and soil conditions, which help it spread rapidly across forest, grassland, and lands. Ecological impacts is biodiversity loss, soil nutrient imbalance, decline in forest regeneration. Need for eco-friendly integrated management, carefully controlled herbicides applications, use of beneficial microbes and degrade plant toxic or suppress lantana growth, phytoremediation plant to restore soil quality and ecological balance. Why eco-friendly approaches to synthetic herbicides alone, resistance development, soil toxicity, mechanical removal, expensive and often ineffective. Needs and stable long-term management, integrated method reduce environmental impact and improve soil health. Biology and speed of lantana camera. So first is reproductive through seeds, and second vegetative propagation and allopathic property. Fourth is a formation of dense thickets. Herbicides based eco-friendly management. Common eco-friendly herbicide used, for example, glyphosate,
आरती जी, आप
1 to
स्लाइड पढ़
2%,
रहे
and
हो,
trichlor.
आप कंक्लूड करो ना, आपका रिसर्च क्या बता रहा है.
यस मैम. मैम मेरा रिसर्च लैंटाना कैमरा के ऊपर है, जो अभी के डेट पे एग्रेसिव
Anand Mehta, Sushil Kumar Patel, Dipti Gupta, whosoever present here can start the presentation. Sailesh Kumar Kumbhar, Selvi Thul, Vineet Kumar,
Yes, ma'am.
okay, you can start, sir.
Vineet
Hello.
ji, please turn on your video and start the presentation.
विनीत कुमार.
जी
विनीत कुमार.
जी सर, थैंक यू सर. आई विल शेयर कर रहा हूँ सर मैं. जी
शेयर कीजिए प्लीज.
जी
सर.
You can start, sir.
स्क्रीन से शेयर होगा मैम.
Screen share. There is an option of screen share, sir. You can start from there. Otherwise, you can give the presentation orally. No problem, sir.
Okay, ma'am. Thank you. Good afternoon, everyone. My name is Vinit Patwa, research scholar, Dr. C.V. Raman University, Bilaspur. My topic is impact of environmental changes on Hymenoptera in Chhattisgarh. मैम, जो हाइमेनोप्टेरा होता है, इसके अंतर्गत में हनी बीज आते हैं, एंड्स आते हैं और वेब्स आते हैं। ये इकोसिस्टम में काफी महत्वपूर्ण भूमिका निभाते हैं। लाइक ये जो हनी बीज होते हैं, ये गुड पॉलिनेटर्स होते हैं। जो वेब्स होते हैं वो गुड पेस्ट कंट्रोल करते हैं, और जो एंड्स होते हैं वो इकोसिस्टम सर्विसेज का कार्य बहुत बखूबी करते हैं। वर्तमान में छत्तीसगढ़ में देखा गया है कि तीव्र मानव गतिविधियों के कारण हमारे जो हाइमेनोप्टेरा कम्युनिटी है उस पर बहुत गंभीर प्रभाव पड़ा है, इसलिए इसका अध्ययन करना आवश्यक है। मेरा जो इसका मेन उद्देश्य है कि इन्वायरमेंटल चेंजिंग का हाइमेनोप्टेरा की डाइवर्सिटी और डिस्ट्रीब्यूशन पर प्रभाव का एनालिसिस करना है। निष्कर्ष ऐसे बताते हैं कि छत्तीसगढ़ में इन्वायरमेंटल चेंजेस का काफी बड़ा इफेक्ट पड़ा है। जैसे आवास का विनाश हुआ है, तापमान में वृद्धि, वर्षा पैटर्न में बदलाव, पेस्टिसाइड का अधिक उपयोग, औद्योगिक अपशिष्ट के कारण हाइमेनोप्टेरा की जनसंख्या में काफी गिरावट देखने को मिली है, जिसके कारण एक क्षेत्र का नाम है कोले पड्डी वेट्सलैंड। जैसे यहाँ पेस्टिसाइड का काफी अधिक मात्रा में उपयोग हुआ था, जिसके कारण बीज हैं उनकी संख्या में भारी गिरावट देखने को मिली। और इसका सीधा सीधा प्रभाव जो होता है वो कृषि उत्पादकता और पारिस्थितिक संतुलन में देखने को मिलता है। सरकार द्वारा अभी वर्तमान में बहुत सारे सराहनीय पहल किए जा रहे हैं, लेकिन इसके बावजूद भी और कुछ सराहनीय पहल किए जाना आवश्यक है, ताकि जो हाइमेनोप्टेरा के कम्युनिटीज हैं इनकी समृद्धि बनी रहे और आगे के लिए हमेशा के लिए सुरक्षित रह सकें। थैंक यू मैम।
थैंक यू सर, थैंक यू।
ओके,
Thank you very much, sir. Next participant. And I request you all, please don't wait for your name to call, as we can't see your name here, botany and all. So I can take the name. So please be ready with your presentation and give it as soon as possible. Next, whosoever is present, like Sailesh Kumar, Selvi Thul, Gauri, Dipti Gupta, Aarti Sahu, whosoever want to present the Dipti Gupta, do you want to present?
And the generalization of industrial and municipal wastage and bio-wastage waters. She is currently a researcher at AMHBI, Cape Peninsula University of Technology, South Africa, where she developed nature-inspired, resource-efficient system for tertiary treatment of complex effluents, including tannery, winery, distillery, and other agro-industrial wastewaters, with strong expertise in microbiology, algal biotechnology, and water engineering. Dr. Ranjan has significantly advanced microbial bacterial system for nutrition removal, biomass valorization, and circular bioeconomy application. Her research appeared in reputable journals such as Algal Research, Biomass and Biorefinery, Applied Energy, and others. She also contributed book chapter to internationally recognized publisher, including Elsevier. Dr. Ranjan has presented her work widely at international conferences and is frequently invited as a keynote speaker and domain expert on bioremediation, waste valorization, microbial constria, and eco-efficient wastewater treatment. Her broader scientific interests include microbial interaction, fermentation, microbial, bioplastic, wastewater engineering, biomass to biofuel process, and integrated bioprocess development for sustainable environmental management. During her doctoral study at IIT Guwahati, she earned the JRF and SRF fellowship from the Ministry of New and Renewable Energy, Government of India. Later, she has been awarded the prestigious Erasmus Fellowship for her postdoctoral research at UPB, Spain. She has also served as a DST Research Associate Women Scientist at the National Academy of Sciences, India, Allahabad, contributing to national scientific advancement. A committed mentor and collaborative researcher, Dr. Ranjan works closely with academic and industry partners to translate laboratory innovation into real-world environmental solutions. Her work is driven by a passion for harnessing microbial intelligence to develop greener, scalable, and cost-effective technology for sustainable future. She will connect with us through online. Welcome, ma'am.
Dipti, are you ready?
Yes, sir, I'm ready.
Okay, please turn on your video and start the
Sir,
presentation.
if you allow me, I'll also share my screen. I'm ready with my PPT.
Yes.
Allowed.
Sir.
Yes, you are allowed. Just select you. We are seeing your PPT. Yeah.
Okay.
No. Again, it was disappeared.
This stress was given against the controlled BBM media. So out of my experiment, glycerol has performed the best. It has given around 10.5% increment in the total lipid content, followed by sucrose and sodium bicarbonate. And apart from this, glucose, and sucrose has also led to a morphological changes that can be seen as in the control condition. The sample was in same morphological state, but after 10 days and after 30 days, it changed its appearance to a yellow color. The filaments were not green; they changed into yellow color. So these two were my conclusions. And if we supply glycerol along with BBM media, we can extract more of the lipid content, and it will be helpful for the biofuel extraction in future.
Okay, thank you.
So it's all I have. I have compiled my presentation.
Ma'am.
So should I mail my PPT to you in the group, or I have to mail the PPT to the same ID that was given? Hello.
Yes, ma'am. Please mail us.
Okay, sir. I'll just mail the PPT.
Okay, ma'am. We will share that PPT from here.
Okay, sir. Thank you so much. This is all.
Ma'am, please tell me in which mail you are going to send.
Sir, I'll send on the same ID that we have sent our abstract IDs.
Okay, ma'am. D.L.S. International Seminar. Okay,
Yes,
ma'am.
sir.
Please do.
On the same mail ID, I'll send my presentation also.
Yes, ma'am. Yes, ma'am. Please continue.
Okay, so a very good afternoon to everyone. I'm Dr. Amrita Ranjan, and unfortunately, I was not able to hear when I was introduced, but I would just like to tell a little about me that I'm working as a researcher at Applied Microbial and Health Biotechnology Institute at Cape Town right now. And my research is very much focused on the bioremediation and valorization of various waste and wastewater from different resources. And the resources can be agricultural waste or the municipal waste or various kind of industrial waste. Today's presentation is basically on the microbial route for the bioremediation of the wastewater. So before we start, let's have a very quick overview. We will start with discussing a little about the global wastewater challenges, and then we will talk about the wastewater, which basically I'm dealing with. And for this presentation, I have selected tannery wastewater. And for the remediation purpose, as I'm following the microbial route, we will discuss my research inputs in terms of microalgal and microalgal bacterial consortium for remediating the tannery wastewater. We will discuss in a very short about how the experiments were performed and what was the output and how we try to analyze the product through it. And finally, we will also talk about the challenges and the scopes and the future work which is related to the study. So let's start with the global wastewater crisis. Globally, the wastewater sector is under severe stress. Over 80% of municipal sewage remains untreated, and only 38% of industrial effluent undergoes the safe treatment, leading to a sustained pollutant loading not only into the existing water systems, but also to the land and the other environmental factors. So consequently, aquatic system accumulates reactive nitrogen, phosphorus, high COD, and heavy metals, which is driving towards the eutrophication and also leads to the toxicity and overall degradation of water quality. So these untreated flows also pose direct public health risk and increase the water scarcity, as the contaminated water is not actually available for the domestic use or agricultural use or for some other industrial purposes. So here comes the importance to actually remediate these effluents which are coming through these different sources, which are highly polluted and highly loaded with organic and inorganic pollutants. And also from the climatic perspective, the anaerobic decomposition in unmanaged wastewater streams contribute roughly to 1.57% of global greenhouse gas emissions. So primarily these gases are methane and the nitrous acid, but also includes the other, which we cannot actually ignore. Although these pressure creates a fundamental barrier to achieving United Nations Sustainable Development Goal, the SDG, which is more focused towards this goal, is the SDG 6, which explicitly target wastewater treatments, its safe reuse, and the protection of the water bodies. Today, in this presentation, the wastewater we will be talking about is the tannery wastewater. And in Cape Town, we have certain tanneries, and the effluent that comes out of these tanneries are heavily loaded with lots of organic and inorganic pollutants, and this needs to be treated. So this is where the actually industries contact us as a researcher, and they seek help to remediate these wastewater so that they can be safely disposed into the nature. These tanneries, they also put efforts to remediate these wastewater to certain extent, and generally the primary treatment and the secondary treatment are done within the tannery itself. The generated effluents are actually developed after several processes of leather processing, which is like soaking, liming, de-liming, pickling, chrome tanning, and so on. And resulting effluent are actually characterized with high salinity, alkalinity, organic load, and suspended colloids. It also contains a good amount of protein, fat, sulfides, ammonium, chloride, and other components, which, and in addition to this, it also contains certain heavy metals which are actually toxic if they get further oxidized, like the chromium 3. Under certain environmental conditions, it gets oxidized to chromium 6, which is very toxic, and we don't want such oxidation happening into the nature and going to our water bodies and affecting not only the aquatic plants but indirectly affecting the humans also. So, and the major problem with tannery wastewater is it is highly variable. Every batch that comes out of the tanneries is actually very different from the first batch. It is not only because of the processing, but it is also, this inconsistency is also sometimes seasonal dependent, and sometimes some of the units doesn't work that perfectly, and that leads to the discharge which is very much different from the previous batch. So these are the challenges when the batch you are receiving from these tanneries, they are not uniform, they are very different from each other. The challenges is to develop a method that should actually work for all the batches you are receiving from these tanneries. Now let's talk about the treatment of this tannery wastewater. The primary treatment happens generally in the tannery itself, and it majorly removes the bulk solid, hair, fat, colloids in tannery wastewater through screening and pH corrections, coagulations, and the settling processes. Sometimes the secondary also takes place within the tannery depending upon the infrastructure of tannery. Otherwise, we bring them to the lab and does the ex situ treatment. And it majorly includes the biological processes like activated sludge, sequential batch reactors, to break down the dissolved organic ammonia sulfide, and this produces a comparatively much clearer effluent than what actually initially it was. Now, even after this secondary treatment, the water which comes out, or the effluent, is not ready to get safely discharged or can be reusable, because of such a higher organic and inorganic load. So it needs a further treatment, which we call it as a tertiary treatment. And this tertiary treatment is where we actually comes up. And in this case, for today's work which I'm sharing with you, we will be discussing about the microalgal and bacterial consortium use for treating the secondary treated effluents from the tannery industries, so that they are remediated very well and are ready to get disposed of in the nature safely, supporting a safe environment. And also, while doing so, we tend to utilize the biomass which we produce during the whole process, using that biomass for the valorization purpose that generates certain value added products. And this is how we contribute to the circular economy of the whole system. Before we start any experiment, the first thing is to know the water quality which we are receiving. And as I've already told, each batch is very distinct from the other batch. Like here, for an example, I have compared two batches, and you can clearly see that both the batches are very distinct. The first batch, you can see, is very nicely treated at the tannery itself, while the second batch, maybe because of certain failure at certain stages, is highly loaded with organic and inorganic components. So you can see a very high COD and TOCs and ammonium content for the batch 2, while comparatively lesser batch for the batch 1. Although, not to forget, both the batches are still not ready to get safely disposed into the environment. So the tertiary treatment is needed for both the batches. But now here is the place where the challenge comes. The challenge is, comparatively, it is easy to grow certain microbes in effluent which is not strongly loaded with say organic and inorganic content, but a effluent which is highly saline, which has a very high COD, which is having very high ammonia, phosphorus, and sulfates and sulfides, microbes tends not to grow that beautifully in such effluents. So the idea is to develop a microbe or microbial culture that can actually grow well in all the batches which you receive from the tanneries, irrespective of the concentration of the organic or inorganic pollutants in it. So now as a microbiologist, if you see, we will see that right now we have talked about the tannery wastewater, which is basically a feedstock for our cultures. And now we will talk about the culture we are dealing with. Today we will be dealing with the microalgal and bacterial consortia, where we are growing microalgae as well as the bacteria together. And we will try to remediate the wastewater in a way that it can handle and reduce the pollution level from the effluent. So microalgal bacterial system is a very beautiful system because it has a natural synergy between algae and bacteria, making them highly suitable for complex tannery wastewater. Microalgae photosynthetically generate oxygen, which support aerobic bacteria in breaking down organic matter, oxidizing ammonia, and reducing sulfides. In return, bacteria releases carbon dioxide and nitrogen and other nutrients that helps and stimulates the microalgal growth. So this mutual exchange allows the consortium to function efficiently even under very saline and turbid and metal-rich tannery wastewater. Let's discuss this microbial bioremediation route. So as you have now understood that we are dealing with microalgae as well as bacteria simultaneously, and they are growing in synergy for the system. So in wastewater treatments, these microbes employ several biochemical pathways to remove pollutants. Some of the routes are like biooxidation, biodegradation, which is generally done with bacteria, and that includes the enzymatic breakdown of complex organic contaminants such as phenols, dyes, hydrocarbons, and tannery derived organics that are actually broken into the simpler or less toxic products like carbon dioxide, water, and biomass. Next comes is phycoremediation. Phyco is basically for the algae. So now what does algae do? So algae actually does the assimilation of nitrogen and phosphate for growth, producing oxygen through photosynthesis to support bacterial degradation and capturing metal through cell surface functional growth. So this makes algal system particularly efficient in nutrient-rich or saline wastewater. Now let's discuss a little about the biorefinery, which we are handling here at MB. So what happens is we receive the influent from the tanneries. We have we have a huge group, and each group has its own expertise. So one of our expertise group, they are expert in generating this HLFCR reactor, whose primary objective is to remove sulfur, and they can harvest the sulfur out of it. Removal of sulfur is very important because that reduces the toxicity level of the wastewater, and once it has undergone through that process, then we send it to our anaerobic digestion unit, and from where we get biogas along with the sludge. Once it is treated through the anaerobic digester, the wastewater that comes out is now secondary treated, and we take it to our algal system to grow our algal system. And as a result, in presence, whether at the flask scale or at the reactor scale, we receive the water which is highly remediated and is safe for the disposal into the environment. At the end of the whole process, we receive algal, or more accurately, algal bacterial biomass, and we further process it into say biodiesel by transestrifying the lipid content of it, or if it is very rich in protein, we go for the animal feed, and if it has a good NPK ratios, then we go for utilizing it as a biofertilizer. Sometimes these are also rich in carbohydrate, so we have also tried to check its potential to produce bioplastics. So depending upon the final characterization of biomass which we receive at the end of the whole process, we select its fate. So now at MB, how the process start? It's a very short and quick overview. Before starting before coming to consortium, we try to screen and select the microalga which has the potential to grow in such a highly loaded and saline environment, because not all microorganisms can actually sustain in such heavily loaded systems. So what we have done is, we have selected some indigenous microalgal species. We have isolated from which is from the periphery of the tanneries only, which has the potential, or which has shown the potential to grow in such tannery wastewater naturally. We have screened them. We through the molecular studies, we have actually identified them, and after the strain selection and identification, we have tried to acclimate them into various concentration of tannery wastewater, starting with say 10% tannery wastewater. Once they are acclimatized, you go to next 20%, and slowly you go to the 100% of tannery wastewater. And it really takes time. It nearly takes 8 months to check which strain can actually withhold this kind of extreme environment and can still produce some good amount of biomass that can be of your use. And once we have selected our strains, then we go for the MAB studies, because these are practically more more observable, because in natural environment, if you from flask, when you come to the reactor, and then from reactor you go to the open ponds, you cannot just omit the bacteria there. So bacteria will always be there. You can just try to reduce its number, but they will be there. And it also assists in the remediation a lot. So if they are there, they're actually helping you. So finally we go for the MAB studies with these selected microalgal cultures, and then we do the data collection and model generation. And once the flask studies are optimized, then we go for the reactor studies, and we try to scale up at the reactor scale. This is the reactor which we are using in our department, and this is a huge reactor. Huge reactor in terms, it can take 20 to 25 liter of the tannery wastewater. We do all our experiments in duplicates to confirm the reproducibility, and to be sure about what the results which we are receiving is consistent. It is an automated system where we can actually maintain the temperature, the photo period, the light intensity, the shaking, and even we can monitor the pH and oxygen and DOs. So everything is built in there. So as I have told you about the selection and identification, this is a quick overview of that, where we acclimatize tannery effluent. We acclimatize our microalga with our tannery effluent. As I've told, it takes nearly 8 months. And finally, we have selected 3 indigenous strains, which we have found in our nearby region, and 1 commercial strain, which we have procured from the CCAP UK. And all these 4 strains, they exhibited a very halotolerant nature, and they have been successfully grown in 100% of tannery wastewater. So once we selected them, we have done their molecular identification using the primers, the algal primers, and we have also done the microscopic confirmation using the SEM as well as the confocal microscopy studies. So finally, the 4 strains which we have selected is basically Tetradesmus obliquus, Tetradesmus dimorphus, Tetradesmus obliquus, another strain, and the Neochlorus. The remediation potential of these were checked at the ambient condition, which is 21 degrees Celsius, along with the shaking at 150 RPM, and the photo period was kept at 14:10, which is light to dark cycle. The selected strains were then studied for their growth profile to understand how they grow into the in tannery wastewater. And all strain exhibited a rapid early growth phase, but Tetradesmus obliquus strain consistently achieved the highest biomass, indicating strong tolerance to the harsh tannery wastewater matrix. The mixed consortium, as you can see here, the mixed consortium also performs well initially, reflecting complementary interaction in the contrast. Neochlorus initially grows slowly and shows a little lesser adaptability, but the overall, the profile highlight strain specific resilience and the potential advantage of the selective consortia for stable biomass production in tannery wastewater. Here, these graphs, they compare how each strain influenced carbon and nitrogen removal by the end of experiment. For COD, all strains show an apparent increase in the COD, that is total COD, relative to the starting level, mainly due to biomass buildup and particulate release, while the soluble COD changes are minimal. In contrast, the total organic carbon increases modestly, but all strains consistently reduce total nitrogen by 25 to 36%, showing effective nitrogen uptake despite the complex tannery wastewater matrix. The dotted lines, they highlight the initial concentration, making the shift in each parameter easy to see. Now, the next step for the remediation is the ammonia. The ammonia concentration decreased from relatively low concentration in tannery wastewater to below the detection limit at the end of the test for all the strains over 13 days. During rapid ammonia utilization, these were accompanying increase in pH and decrease in total alkalinity. Increased alkalinity after 4 days was attributed to fixation of carbon dioxide and possible proton uptake. This increase in total alkalinity from microbial growth is a very good example of how microalgae and bacteria can operate synergistically in amoebic system, where microalgae can provide the alkalinity required by the ammonium oxidizing bacteria. So we can see that the selected microalgae strain all showed promise for polishing tannery wastewater after treatment after primary and secondary treatment in terms of ability to grow in tannery wastewater utilizing ammonia. The result of this study shows that microalgae require bacteria to synergistically increase COD and ammonia removal. Once the monoalgal culture and the algal consortium study is optimized, and we know what consortium is the best and what are the conditions in which they can actually give some good remediation, then we come to check the seasonal influence of this remediation using the consortium. So for that, we all the studies are done using statistical softwares. We design our experiments using statistical software like Minitab, Statistica, or Design Expert. That gives a good layout of design of experiments. And for this seasonal influence, we have selected three variables. The first one was the season, and the others were the initial ammonia concentration and then the inoculum size. The season, we have selected three levels for season, which is summer, spring, and winter. And the data which we have received here is the average data, which is actually seasonal data, which is observed in Cape Town. So you can see the temperature and the light. This is all a mimicking of actually what happens during those seasons in Cape Town, so that when we scale up our optimized conditions into the much bigger scale, then when they are exposed to certain season, we know what to expect from these consortium when they are growing in tannery wastewater. We have the figure shows that this total COD increase in all flask when it comes to the seasonal effect. And this is very consistent pattern observed also in the single species culture. And unlike total COD, soluble COD comparatively remains stable. And the total COD is peaked under the summer condition. You can see the summer condition, it has the peak. And this signifies that the biomass was the highest during the summer conditions. And this is what we actually needed, because the higher the biomass, the higher will be the valorization potential and also the bioremediation effect on the tannery wastewater. This is the characterization of biomass. So at the end, when we have received the biomass and we have remediated our tannery wastewater successfully using the tannery wastewater and using the microalgal and bacterial consortium, and the levels of ammonia has come down nearly to zero, what we do is we take out the biomass, we dry it overnight, and then that dried biomass is then characterized to know its organic and inorganic content, to know its carbohydrate, protein, and lipid content, so that we can decide its further fate. So like in this case, you can see that the carbohydrate as well as the protein content is comparatively good, while the fatty acid is not that good. So this indicates that when we want to valorize our biomass under these optimized conditions, uh, it is not very good or promising for the biodiesel production, because it has a very low lipid content, but because it has some good carbohydrate content, it can be further microbialated to produce bioplastics kind of stuffs. And the protein part and the higher NPK ratio make them efficient as to be used as a biofertilizer or as a protein supplement or the feed for specifically for the cattles. And definitely at the end, the best thing which we have been looking for is the effluent which has a very safe organic load and is now can easily be disposed of in the environment without any further polluting the environment. Now, all looks good, but there is whenever you perform a research, there is always certain limitations and constraint with which we always fight while doing the research. And as we have already discussed, the high salinity and hardness of the tannery wastewater, the toxic metal which is there, the colloidal and opaque nature of wastewater, which makes many of the spectrophotometric analysis nearly impossible. And then you look for other alternative possibilities of checking the content, or say checking the growth pattern, which could be like say gravimetric. And then variable wastewater composition, every batch, as I have said, differ from each other. So making something, making a consortium that works with all the batches is a challenge in itself, which we have achieved to a certain extent from our long experience in this area. And it is sometimes, depending upon the algal cultures you are using, sometimes it becomes difficult to harvest the biomass. We are lucky in our case because the Tetradesmus neochloris and the neochloris cultures which we have selected as a consortium, they tend to make a cynobia. Cynobia is like an agglomerate where after certain concentration they achieve in the wastewater, they tend to accumulate and then they settle down. So because of this characteristic of making cynobia, it's very easy for us to harvest them. We just at the lab scale, we just centrifuge them, and then you can take the pellet and dry it overnight under the suitable conditions of temperature, and the biomass is ready to measure for the characterization. There is certain more when you scale up the process, like you start with 100 ml, then you go to 250 ml flask cultures, then you go to 500, then a liter, and then after a liter maybe then you go to this reactor scale where you are handling a 20 liter of tannery wastewater together. Every step is not similar to what you have done before, because when you scale up, the conditions change. Just for a quick example, I can give you that when you perform an experiment at 100 or 250 ml flask, the light penetration is very easy. But when you perform the same with a reactor where you're having 20 liter, because of the colloidal nature of the tannery wastewater, and once the algae tend to grow more to the light doesn't pass through clearly, right? And the light sources are outside, so the algae try to grow more towards the wall rather than towards the center. So then you need to think about adding certain factors that makes them off the wall, and that can allow more light to penetrate so that the photosynthesis keeps on happening and the alga doesn't die. Not to forget, you are not only aiming to grow alga, but you also want your bacteria to go there, because those are the ones who are doing the nitrification and denitrification. So managing pumping of water, managing the pumping of air inside, making certain pockets which are anaerobic zones, and it's important for growing the denitrifiers there. So it may sound easy, but when you start performing it, optimizing the whole thing, it takes time and it's very challenging, and you need to monitor it on a daily basis to see everything is actually going right. Future direction, definitely research never ends. You can always accelerate it. We are looking to develop a stress tolerant MAB consortia, and we always look forward to improve our reactor designs. We we also tried to apply proteomics and metabolomics for the real-time monitoring. And we also want to work with some other algal cultures, and maybe we want to enhance the biomass harvesting when we are scaling up the process, because you don't want to use centrifuge when you are using say 20 liter or 50 liter of reactor. So, and the most important thing is to generate value added products in terms of high biomass, so that we can contribute significantly to the circular economy and make the process more sustainable. And this is how the whole biomass can be converted into bioenergy, or say of more industrial use. And then finally integrating MAB system into the circular economy pathway for tanneries. So this was all from me. And I would like to acknowledge definitely the conference committee for giving me this opportunity to share my research with you all. And definitely my colleagues and my team at MB, Cape Peninsula University, my collaborator from University of Cape Town, and the funding bodies here, which is Water Research Commission, for making this research possible. These are the references from where you can see my work. These are the recent ones, including the report, which the WRC report, which has all these details. And thank you so much. And let's contribute to a better planet together. Thanks a lot.
Thank you very much, ma'am. Am I audible?
Yes, Neha, I can hear you.
Yeah, thank you so much, Amrita. Thanks a lot. It was a great talk from you. And there were many technical glitches and many technical problems. Then too, we managed to connect to you from South Africa. It's been a great. Thank you so much. Looking forward to such more cooperation from you.
Thanks a lot, Neha, and the complete college for organizing this amazing conference. And I really enjoyed it. There is always some sort of technical glitch, but nothing can stop us. I'm also looking for further collaborations. And good wishes to you all.
Thanks a lot. Thank you.
Hello, good afternoon, ma'am. Myself, Sushil Patel. I am ready, ma'am.
Good afternoon. Just wait a minute, sir. Actually.
Okay, ma'am, no problem.
Actually, we are having our next resource person online. So,
okay.
Hello. Yes, you are audible, ma'am.
Hello. Hello. Am I audible?
Yes.
Myself, Dr. Ramakant from Chaudhary Johnson University, Meerut, Botany Department. May I start my presentation, my lecture?
Yes, sir. Yes.
Okay. सर्वप्रथम तो मैं ऑर्गेनाइजिंग टीम डॉक्टर अर्चना पांडे और उनकी पूरी टीम को धन्यवाद देना चाहूंगा, जिन्होंने मुझे इस अपॉर्चुनिटी को आप सबसे इंटरेक्ट करने की ये अपॉर्चुनिटी मुझे उपलब्ध कराई. Already you have enjoyed the lecture of Amrita Ranjan on phycoremediation or bioremediation of 10D water, 10D waste. My lecture is something related to that one, but it is something different because I have here some different types of algal stains which are capable to biomine of the nutrients from different wastewater, different types of the wastewater. My slides are visible. Hello. My slides are sharing. Hello. आप लोग मुझे सुन पा रहे हैं?
Yes, sir.
Yes, sir.
मेरी स्लाइड्स आपको दिख रही हैं?
Yes, sir. Yes, sir. It is visible, sir, and you are audible also.
Okay, okay.
You can continue,
So the
sir.
title of my topic is Nutrients Biomining from Wastewater Using Algae. As you know already, we are facing a lot of challenges regarding the pollution in different habitats or in the different systems. So water, as it is considered an essential element. If you'll go according to तुलसीदास जी की रामायण आप लोगों ने पढ़ा होगा, तो वो कहते हैं कि छित जल पावक गगन समीरा पंच तत्व यह रचित शरीरा। यानी शरीर जो है, जो ऑर्गेनिज्म है, उसकी शरीर लगभग इन 5 तत्वों से बनी होती है। उनमें से एक तत्व है, प्रमुख है, जिसे कहा जाता है जल, और जल ही जीवन है। यानी विदाउट वाटर वी कैन नॉट सर्वाइव। कोई भी ऑर्गेनिज्म बिना जल के नहीं रह सकता, वाटर के बिना। बट द प्रॉब्लम इज हियर व्हाट वी आर फेसिंग टुडे, बिकॉज मोस्ट ऑफ द हैबिटेट्स or water bodies are polluted presently. Particularly, it is very bad situation in India. Most of the rivers, including Ganga, Yamuna, and so many rivers of the country, even Narmada, and so many rivers are heavily polluted by different types of the pollutants because of so many regions. So water is one of the most important natural resource. It affects the vital processes of all living organisms. Water covers more than 70% of the Earth's surface. In nature, water is available to plants in 3 forms. They are atmospheric moisture, or in the form of precipitation, or from soil water. Further, the quality of water is also important, especially for aquatic organisms. The challenges faced by the organisms. Organisms face challenges due to heavy pollution and contamination in water bodies. As I told you, these pollutants are released by industries and so many untreated wastewaters or sewage treatment plants, which directly release to these different water bodies, particularly rivers. The Ganga, even the river Ganga, holy river Ganga, faces a significant water quality challenges due to pollution from various sources, including industrial waste and domestic waste. I'm not going in details of the pollution, but what are the conditions? You can see here impact of pollution on the rivers. You can see here, हम लोग सनातन धर्म को मानने वाले लोगों में से हैं, और हम लोग लगभग हर प्रकार की जो पूजा पद्धति, तमाम यज्ञ आदि करते हैं, स्नान दान करते हैं, वो लगभग नदियों के किनारे या नदियों में डुबकी लगा करके, और ये हमारा एक प्रमुख पर्व माना जाता है उन्हीं में से। तो देख सकते हो यहाँ पर ये जो डिफरेंट कंडीशंस हैं, डिफरेंट रिवर्स हैं, जहाँ जिसके सामने द पीपल दे हैव डम्प्ड a lot of pollutants in the river banks. Sources of pollution. Industrial effluents include industrial discharge, particularly from cities along the river, contribute significantly to water pollution. Domestic wastewater. Here, untreated sewage from cities and towns is a major source of contamination, adding organic pollutants and pathogens to the rivers. Another pollutant is here agricultural waste. Pesticides and herbicides from agricultural lands can contaminate the rivers through runoff, affecting aquatic life and water quality. And last one, that is the religious practices which are being adopted by us. Religious activities, including the use of plastics for offerings, can also contribute to pollution. कोई भी यदि हम लोग कहीं भी रिवर में स्नान करने जाते हैं, तो इस समय सबसे बड़ी समस्या है कि हम कोई भी पुष्प भी लेकर के जाएंगे तो पॉलीथीन बैग में लेके जाएंगे, और उसको भगवान को अर्पण करने के बाद या नदी में चढ़ाने के बाद पॉलीथीन बैग वहीं फेंक देते हैं, उससे भी पोल्यूशन हो जाता है। यदि आप देखोगे रिवर्स इन सनातन धर्म में रिवर की उपयोगिता क्या है, एक श्लोक है जो जिसको मैं यहाँ पर रिसाइट करना चाहूंगा। गंगे च यमुने चैव गोदावरी सरस्वती नर्मदे सिंधु कावेरी जलेस्मिनी सन्निधिम कुरु। प्रायः सनातन धर्म में जब लोग कोई भी स्नान करते हैं तो इस मंत्र का जाप करते हैं और कहते हैं ये उपरोक्त वर्णित नदियां हमारे जो है जीवन को सफल करें और हमको पवित्र करें। The sacred rivers of Sanatana, the 7 sacred rivers of India known as Saptarshi in Sanatana, and these are Ganga, one of the holy river, the most revered river believed to wash away sins, पाप को धुलने वाली। Yamuna, another important river in Hindu mythology, Narmada, known for its westward flow and significance in the western region of India. Godavari, known as Ganga of the south, Saraswati, an ancient river mentioned in scriptures, currently believed to be an and Kaveri, a sacred river in the southern part of the India, and Sindhu, Indus River generally known as one of the largest river in the world with historical and spiritual importance. These rivers hold immense spiritual and cultural significance. Spiritual significance of the Ganga. According to Tulsidas जी क्या कहते हैं अपने रामचरितमानस में, कहते हैं कि माघ मकर रवि गति जब हुई, तीर्थ पति आव सब कोई, देव दनुज किन्नर नर श्रेणी, सादर मज्जह सकल त्रिवेणी। यानी अब कल्पना कर सकते हैं कि उस गंगा नदी जिसकी हम पूजा करते हैं, उसके जल की, उसको हम लोग जल बोलते हैं, गंगा का पानी नहीं बोलते, जल, उसकी कितनी बड़ी उपयोगिता इन्होंने बताई है तुलसीदास जी ने। और बताया है कि उस समय माघ में जब सूर्य मकर राशि में आ जाता है तो लगभग पूरे विश्व के देव धनुष किन्नर आदि सभी लोग आते हैं और सब लोग त्रिवेणी में स्नान करते हैं। इसकी उपयोगिता बताता है। यहाँ पर एक देख सकते हैं कितना बड़ा कांग्रेगेशन, यहाँ पर एक कुंभ, महाकुंभ के नाम से जो माघ में लगभग होता है। कुंभ तो लगभग 12 साल में होता है और उसके बाद बड़े कुंभ भी होते हैं, 36 साल में भी होते हैं। रिसेंटली वी हैव आल्सो सेलिब्रेटेड महाकुंभ इन 2025। आप वहां की भीड़ देख सकते हैं कि लगभग कई देशों के बराबर आप देख सकते हो, जितनी पॉपुलेशन होगी उनसे ज्यादा पॉपुलेशन वहां पर महाकुंभ में स्नान किया। और किस महाकुंभ में, वो महाकुंभ होता कहां पर है, लगभग यदि आपको सबको पता होगा कि लगभग जो कुंभ है, कुंभ का आयोजन भारत में 4 स्थानों पर होता है। उन्हीं में से एक प्रमुख है संगम तीर्थ में होता है। यह मेले की धार्मिक और सांस्कृतिक महत्व को दर्शाती है। जब सूर्य मकर राशि में प्रवेश करता है तो महाकुंभ का आयोजन किया जाता है, जो एक महत्वपूर्ण धार्मिक आयोजन है। इस दौरान बड़ी संख्या में लोग पवित्र संगम में स्नान करने के लिए आते हैं। The largest celebrated once every 12 years at the Prayagraj on the confluence of the Ganges, Yamuna and Saraswati, a major pilgrimage and festival in Hinduism occurring 4 times in 12 years at different locations in India. It attracts millions of the pilgrims for spiritual purification. One of them, Haridwar on the bank of the Ganges, Prayagraj on the confluence of the Ganga, Yamuna, mythical Saraswati river, Ujjain on the banks of the Chipra river and Nasik on the banks of the Godavari river. ये 4 नदियों के स्थान पर, इन 4 नदियों के किनारे ये 4 स्थानों पर कुंभ का आयोजन होता है। आप यहाँ पर देख सकते हैं एरियल व्यू अगेन, इट वाज टेकन on February 16 in 2025, the largest congregation in the Prayagraj. But a news was appeared in the newspapers. Water quality at Mahakumbh found unfit for dip on the many days, according to news of Hindu. ये जो न्यूज पब्लिश हुई थी हिंदू में 22 फरवरी 2025 को पब्लिश हुई थी। उसी पर लोग शुरू कर देते हैं कि गंगा रिवर इतना ज्यादा पोल्यूटेड है, वहां पर स्नान नहीं किया जा सकता। तमाम देश विरोधी ताकतें हैं जो उसको और प्रचार प्रसार करती हैं, उसको बढ़ाती हैं और भारत को बदनाम करने की उनकी एक साजिश होती है उसकी वजह से। लेकिन हम मान सकते हैं तमाम गवर्नमेंट एजेंसीज उसका एनालिसिस भी करती हैं टाइम टू टाइम, और उन्हीं में से कुछ न्यूज बाद में एक पब्लिश होती है जो बताती है कि ए रिपोर्ट पब्लिश्ड इन फरवरी 25 by the Central Pollution Board submitted to the National Green Tribunal states that the river quality in the Prayagraj during the Magh Kumbh was fit for bathing. फिर कंट्राडिक्ट एक दूसरी रिपोर्ट आती है 28 फरवरी को, वो बताती कि नहीं वाटर फिट था, गंगा जल फिट था नहाने के लिए। और लगभग ओवर अप्रोक्सीमेटली 55 crore people have visited the Mahakumbh and they have taken the holy dip. So these are some facts. But according to activities restricted, why these rivers are prohibited? Because of so many activities, one of them. But in our mythology, according to Brahma Puran, our ancient text, the clearly mention how humans should behave on the river banks. Our ancestors understood the importance of the rivers and set guidelines for their preservation. The Ganga's sacred banks restricted for 13 types of the activities, including bathing after using soaps, detergents, cloth washing, water pump dumping, and defecation, throwing discarded flowers, etc. But what we are doing? We are throwing everything in the Ganga, and we are heavily polluting to our these sacred rivers by different ways. A report, the facts about the Ganga Jal, you can see here, the river Ganga flows approximately to 2,525 kilometers through the plains of northern India, forming a drainage basin covering 8,61,404 square kilometers, according to Dow 2001 and Joshi et al. 2009. In 1896, Ernst Haenken, the British bacteriologist, first noted the antibacterial properties of the Ganga water or Ganga Jal against Vibrio cholera, and revealed that Ganga water does not support the growth of many infectious microorganisms. और जो लोग सनातन धर्म में विश्वास करने वाले हैं, यदि हमारे यहाँ किसी व्यक्ति का लास्ट अंतिम समय आ जाता है या वो देह त्याग करता है, तो उसके परिवार में उसके जो स्नेही जन हैं, वो उस व्यक्ति को गंगा जल और सोना, इस तरह से तुलसी, इस तरह के कुछ चीजों को उसके मुंह में प्रवेश करते हैं, उसको खिलाते हैं, जिससे कि उसकी तमाम उपयोगिता है। हम उसको यहाँ पर डिटेल में नहीं जाएंगे, रिलीजियस एक्टिविटी आप लोग उसमें ध्यान से देखिएगा। बट लेकिन गंगा से संबंधित और एक अनदर स्टडी आई वुड लाइक टू शेयर हियर। इन अनदर स्टडी द नेशनल एनवायरमेंटल इंजीनियरिंग एंड रिसर्च इंस्टीट्यूट also reported that as compared to Yamuna and Narmada water, Ganga water contains a substantially greater proportion of bacteriophages, according to Neha et al. And this report is available. Anyone can search this report from the website. It is shown here. The Ganga Jal used as holy water on different occasions by the followers of Sanatana. तमाम आयोजनों में, पूजा पद्धति में उपयोग होता है इसका। और ये गंगा का मैप है जो लगभग ये दर्शाता है कि गंगा कहां से कहां तक उत्पन्न होकर के कहां पर जाकर के वो बंगाल में, यहाँ बंगाल की खाड़ी में वो मिलती हैं समुद्र से, इसको दर्शाता है यहाँ पर। लेकिन ये रिपोर्ट, इफ यू विल सी दिस रिपोर्ट अगेन, द पोल्यूशन स्टेटस एंड वेस्ट वाटर जनरेशन in the India, you can see here different states are showing here by this table too. Status of industrial units, water consumption and wastewater generation along Ganga river basins. Uttarakhand approximately it has 42 industry, and Uttar Pradesh has approximately 687 industries, and they generate approximately 693 million generated wastewater. In Jharkhand, you can see here, Bihar again, it has 13 industries, and Jharkhand has West Bengal again, it has 22 industries, and approximately they have approximately 764 industries on the river banks of the Ganga, on the Ganga banks. The status, but the table 3 is showing here the status of STP installed under the sewage treatment plants installed under the Ganga river catchment basins. You can see here the situation is, you can see here in Uttar Pradesh approximately it has 8, and it has installed capacity of 358, and actually utilized capacity it has 287, and STP not in operation approximately 1. And the report is, I am not here, I could not here update it here. It is the old report, but certainly it has been improved, but still the water of Ganga is highly polluted, contaminated. Then what is the solution? How we can control the pollution of the Ganga river? Actually, if you will see that on the river banks there are so many industries, and they are releasing the different types of the pollutants in the river Ganga. If this water, which is this waste water which is released by the these industries, if they install this, if they biomine the different types of the nutrients from this waste water, then they can minimize, they can contribute significantly to the river Ganga, and by this way they can control the pollution load or pollutants load in the water river, Ganga river. So by algae. Then what is the solution? The solution is wastewater treatment by algae. Already you have enjoyed the previous lecture given by Ranjan. Okay, so production of algal biomass, and they can produce the algal biomass in large scale by using this wastewater, and they can they can produce from the base from this biomass biofuel. They can produce different types of the bioproducts, and they can ultimately produce the biofertilizer, and they can increase the yield of agriculture. And in addition to this, they can also produce value addition in the products. What are the algae? Algae are the duplex organism, as you all are aware about this, because of their different trophic levels. Algae are not to be compared with the viruses, bacteria, fungi. But as far as biotechnological applications are concerned, algae are the most preferable organism among the plants. They evolve oxygen, they fix atmospheric nitrogen, and they are considered as the primary colonizer and play a very important role as ecological indicator. You can see here how we can remove, particularly I have mentioned here two micronutrients which are found too much in the wastewater, and particularly municipal waste and industrial waste, chemical industries. So we can remove this, we can biomine these micronutrients from the wastewater by using by growing algae in different flask, wastewater and open raceway ponds, or in bioreactors, photobioreactors. Because we have different types of algae. Most of the algal groups show various steps of the diversity at the subcellular levels, cell wall, chloroplast, reserve food material, and in flagella. And at the cellular, at the thallus level also they have a large diversity, being unicellular to parenchymatous forms. So these all algae, different types of algae can be used for the cultivation or for the treatment of the wastewater. Here you can see the algal growth in rice fields. The growth of this algal, you can see this algal population is showing here, indicating here that this water, it has too much nutrient load here. Because you can see here, the green algae is growing too much, but which is the more useful algae, that is the photosynthetic and also nitrogen fixing forms. You can see here, they are growing in little amount. So this water quality is also indicating how much this water is polluted, which has been used in the irrigation of this rice field. You can see here again, these are considered as the ecological indicator. These two pots, they are showing different types of the algal growth in nature because of their different nutrients load. You can see here, as you can see here, biodiversity at a glance. According to Giri et al. 2024, approximately algae have 50,000 total algae, 50,000 species. And among the cyanobacteria or the blue green algae, you can see here, they have reported approximately 5,523 species. And these all algae can be used for the treatment of the wastewater. You can see the biomining of the, and by biomining of different nutrients, we can produce different types of the bioproducts. We can extract biocolorants, food, feed. We can use them as fuel, biofertilizer, mariculture, and pearl culture, and pisciculture. The nostoc is used as food material in South America and many countries, other countries like China. We can also produce many secondary metabolites, including vitamins, toxins, enzymes, by using these types of the algae. Pharmaceutical products can be produced. Cosmetics we can also use as in cosmetics. We can also produce nutraceuticals from the algae, and we can also produce pharmacological probes, nutrients. We can also use algae for nutrients biomining, and we can also reduce the pollution from different water bodies. Yes, this one is showing here a photobioreactor, open photobioreactor. If we have a polluted water industry, we can collaborate with the industries, different industries, and we can treat their waters by using different types of the algae. Here this is a model, and here another person is producing a spirulina in their open field, open raceway pond in their house. Here you can see here. So it can give a good solution of the unemploymentness. Any student or any person which is interested to start a startup, they can produce the algae, spirulina particularly, and many other beneficial forms in their house, and they can sell in the market. Yes, you can see here they are selling this. They are collecting the spirulina from their ponds, and here you can see their production. And this is the spirulina. You can see here we have a lot of algal culture collection in our labs, and we have tested so many algal stains which have tremendous potential to biomining from different waste waters. You can see here it is our culture collection, and the different students, they are observing here the different culture collections, different cultures of different algal stains. And you can see here in my lab we have here anyone, and here I have conducted a technology. We are conducting here a training program here, and you can see here technology demonstration for the production of algal biomass using wastewater. And yes, we can produce the algal for different purposes. You can see the polysaccharide rich stains we can produce by this wastewater. We can also produce many algal stains which has a rich amount, which are very rich source of cytoamine, UV screening pigment, and polysaccharides. Yes, these are some algal stains which are growing in the merit adjoining area. You can see here, and they have tremendous potential. I will show you the microscopic slides here. Yes, you can see here a polluted water body. Here, what the people, the dairy industry, they are releasing a lot of wastewater, particularly dairy waste, in this water body, and a lot of algae is growing here in this wastewater, and we have isolated these stains which have tremendous potential of biomining nutrients. You can see here these are the some stains, most of them here filamentous forms. You can see the oscillatoria, lingmia, pharmadium, many forms you can see here, and since they are all have been isolated from different polluted water bodies, you can see here, yes, you can see in the 6th number, you can see here it has tremendous potential of ammonium mining from wastewater. Yes, we also use the production of biomass in the by using waste materials in our roof. Anyone can start this types of the techniques in their open places. You can see, yes, here technology demonstration, and here you can see the biomining from wastewater in Australia in Murdoch University in LG R&D Center, where I have taken a training of 3 months. Yes, what they do, they have collaboration with the industries, and they use pig industries based water for the cultivation of algae, and they ultimately they produce the biomass, and they supply to different industries. You can see here, yes, you can see if we are producing the large scale of biomass, then we can also extract the biofuel lipids from this biomass. If we are producing in some heterocystous forms, then we can use them for the enhancement in agricultural products. You can see here our agricultural yield you can increase by using this biomass as biofertilizer. So with this here, I want to have some here information, and I want to share with you. And I also invite to all of you here. We are going to organize a national seminar, and we cordially invite to all participants in the national seminar on biodiversity conservation and sustainable development. The date and time is here. We have here on 10th December 2025. It will start at 10 a.m. and you can note here my email ID and my mobile number. Shortly we will share the brochure of this conference, and it will be very much useful here. So anyone, any teacher, any faculty member, any students want to participate, they may contact me by this email. With this, I'm going to end here this. As you can see here, I have shared so many forms of algal stains. If anyone who want to collaborate with me, I welcome to start the MOU or some other by this, and we can collaborate for the using of different types of algal stains for the protection of different types of the value products or biofertilizer, biofuel, anything we can use. And because here we have a good algal culture collection. Thank you. And the last, and I would like to thank to our organizers, particularly Dr. Archana Pandey and their team and her team members for inviting me and for giving me this opportunity. Thank you so much.
Thank you very much, sir. On the behalf of whole D.L.S family, I would like to thank you and thanks for the nice talk you have presented, which is really helpful for all the new research scholars and research development programs. And we are very much happy to see the next upcoming national seminar by you.
Yes, sure.
Thank you. Once again, I would like to thank you, sir.
Thank you.
Then meanwhile, I would like to welcome Dr. Jyoti Pandey ma'am from C.V. Raman University. So here the chairperson for this technical session is Dr. Jyoti Pandey ma'am. I would like to welcome you ma'am for this session. Thank you.
Mr. Sushil Kumar, please continue. Okay, Ankita, ask for next candidate.
Yes, sir. I
Yes,
will
Sushil.
Yes.
explain, sir. सॉरी शेयर स्क्रीन नहीं हो पा रहा, लेकिन मैं प्रेजेंट कर रहा हूँ, मैं जो भी अपना प्रेजेंटेशन है. Respected dignities, good evening. Myself Sushil Kumar Patel.
Please conclude your talk within 5 minutes, sir, please.
Okay, only 2 or 3 minutes, ma'am. My topic is impact of agricultural pesticides on butterfly population. Critical review for future studies. Myself Sushil Kumar Patel, a research scholar, PhD, Department of Zoology, C.V. Raman University. This paper examines the pervasive effect of agriculture pesticides on butterfly populations, synthesizing evidence from various studies to elucidate the mechanism through which these chemicals contribute to lepidopteran declines. Agricultural pesticides have significantly, through indirect toxicity, a lot of crop production, but severely affect the non-target species, such as butterfly, through toxicity, habit degradation, and disruptions of ecological interaction. The effect of pesticides used in agriculture are not simply linear, but rather encompass complex multifaceted development, reproduction, and navigation. These cascading effects extend to population level, contributing to widespread the declines. Specifically, studies from demonstrated that insecticide, particularly used in agriculture, are strongly associated with community-wide declines, total butterfly abundance, and species richness, including migratory butterfly species. Research on effect of specific pesticide from molecular level, the metapopulation level, is indeed to comprehensively assess non-target impacts, both direct lethal and indirect lethal. Various pesticide classes, butterfly physiology, behavior, and reproductive success. The review provides basis for the future studies on pesticide impact to butterflies. Thank you so much.
Next participant. Next, who is there? Aarti Sahu, MS Priyanka, Selvi Thul. I think, ma'am, you want to present? Please summarize your presentation. Yes, ma'am, you can start. Unmute yourself. Selvi Pool.
No. Hello.
जी, Selvi.
Hello.
Hello. Yes. Yes, ma'am, you can start, ma'am.
No. Good afternoon, everyone. Myself, Steve. I'm a
Ma'am,
research
please say,
scholar
please,
in
ma'am, say
Department
loudly.
of Biochemistry. Hello.
123.
Am I audible? Hello.
Yes, ma'am. Yes, ma'am. You can start, ma'am. Please speak loudly and you can start, ma'am.
Okay. Myself, Sylvie Poon. I am a research scholar from Biochemistry, Dr. Ambedkar College, Nagpur. And my presentation topic is green synthesis, characterization, and biological activities of silver and copper nanoparticles from Calcareous Jejuntia leaves. So nowadays, the green method of synthesis has become a major focus for researchers as it is clear, non-toxic, cost-effective, and environment-friendly approach. As some of the metals such as silver and copper has a major role in biomedical science its due to as it bypasses harsh and toxicity. The plant Helicopterus gigantea as commonly found in the Westland. The plant belongs to the family Apocynaceae. It has many advantages in Ayurveda, Siddha, and other fields. The synthesis of nanoparticles used by Camptophilus gigantea leaves has many phytochemical properties. The materials we have collected and purchased from Department of Biochemistry, Dr. Ambedkar College, Nagpur. The leaves were freshly washed and cutted and formed in a powder form. The powder was heated at 80 degrees Celsius using a stirrer and was further filtered using Wattman filter paper and was further centrifuged at 1500 RPM for 10 minutes. The synthesis of silver nanoparticles using GNO3. The stock was prepared by 9 millimolars and was mixed with 10 ml leaf extract. The further mixture solution was incubated at room temperature in dark condition. Whereas for the copper nanoparticles, we have used copper sulfate pentahydrate for 0.05 molars and was mixed with 250 ml of leaf extract. And it was incubated at room temperature for 24 hours in dark condition. Further, the nanoparticles were centrifuged 9000 RPM for 10 to 15 minutes and was completely dried and were washed multiple times using ethanol repeatedly. So here is the result for characterization, that is for XRD, where you can see the sharp peaks at 32 P80 degrees. It is the sharpest peak for silver nanoparticles. Whereas for copper nanoparticles, you can see 29.830 degrees. Whereas for FTIR, you can see the formation of
Shelby,
silver
am I audible?
nanoparticles.
Shelby, am I audible?
Yes, ma'am.
Because of some technical issue, I'm not able to hear you. Will you please summarize your work, what you want to do and what you are doing?
So right now I'm working on further plan, that is antimicrobial activities for S. aureus and E. coli.
What is the objective of your study?
That is synthesis and characterization and it is a comparative study silver and copper nanoparticles.
Okay. And then what is the outcome of your study?
It has various biological activities such as antimicrobial, antioxidant, and anti-cancer, which inhibit the growth.
which inhibit the growth?
Microorganisms, that is E. coli and also S. aureus.
Okay. How much is extent it will helps to the public domain? Public domain में आपके काम का क्या implementation हो सकता है?
Help, like it has potential which can do of antibiotics, whereas for antioxidant and anti-cancer activities.
Anti-cancer activities. ओके
Yeah,
तो क्या
it's
आपकी
like
स्टडी में कुछ इन विट्रो स्टडीज भी है, इन विवो स्टडीज भी हैं, इन सिलिको इन विवो?
no, it's in vitro.
In vitro. Okay. Thank you very much.
Thank you.
Ma'am, can I present now Dr. Kumar Gaurav, oral presentation?
Yes, yes.
Can I present now,
Yes, you
ma'am?
can start, sir. Yes, you can start, sir.
क्या नाम है इनका?
Okay, my slide is visible now?
Yes.
Okay. So I am, first of all, I'm thank you for organizing committee to give me opportunity to present my work on this platform. I am Dr. Kumar Gaurav. So today I am presenting the wastewater treatment as with the help of the microbial fuel cell. So microbial fuel cell, this is the type of the bioreactor or electrochemical device. In that one's the chemical energy which is stored in different type of the organic matter and the waste that is converted into the electrical energy. So microbial fuel cell that is consist of the anodic chamber, cathodic chamber, and the proton exchange membrane. So anodic chamber, that is anode chamber, aerobic, that is the cathode chamber, and the transfer of the electron with the help of the proton exchange membrane. So what is the advantage of using this microbial fuel cell? So it can be reduce the operational cost. It can be treat different type of the wastewater. So in the main advantage of that ones, it can produce the electricity as well as the wastewater treatment. So in this one, if we see this is the anodic chamber and that is the anaerobic. So in that ones, the carbohydrate, it decomposes it, it produces the carbon dioxide, hydrogen, and the electron. So this electron is transferred into this anodic chamber, and in that ones the electron is transferred from the external circuit, and here it can be transferred. And the proton which is generated by this decomposition of the organic matter, so proton can be transferred from here. So in that ones it produces the water. So when due to the transport of the electrons, so there is a generation of the electricity. So for that ones, the main thing in that one's the proton exchange membrane that are the costly. So for that ones we can use the Ultrex membrane, Nephron membrane. So in that ones, when you're using the Nephron membrane, they has the costly membrane, and they are the several disadvantage. Main disadvantage of that is the cost. So for that
Sir,
ones,
what is the objective of your study?
ma'am, that is the synthesis of the proton exchange membrane, and we can see that is the electricity generation, and we can see there are the different type of the CO2 reduction, BOD reduction, and the TS, total dissolved solid.
Means the bioremediation of wastewater.
Yeah, bioremediation of wastewater using the microbial fuel cell, ma'am.
Okay, okay. So what the methodology you'll be utilized?
Ma'am, we have used the membrane, proton exchange membrane, and we have used the microbial fuel cell. So in that ones, we grow the bacterial culture we procure from the NCIM, and we grow on the LB agar and anode, what is the graphite and the cathode, and we develop the specific type of the membrane that is the PMMA, that is the polymethyl methacrylate, that is act as the proton exchange membrane, and we measure the current and the voltage with the help of the multimeter. So this is our schematic diagram of the proton exchange membrane. So we have designed that this microbial fuel cell, and we go for the further treatment of the wastewater. So in that ones, the microbial fuel cell, which is cheaper, and in that ones we can synthesize with the help of this schematic diagram. So first of all, we take the polymethacrylate dissolved in the dimethyl formaldehyde, we mixture and stirred the 40-degree uniform solution. After that ones, we added the citric acid as a doping, and we can also add the potassium cyanide. And when adding the citric acid, the concentration 1, 2.5, 10, and we obtain the homogeneous, then we go for the hot over.
Outcome. So ठीक है, methodology आपकी ठीक है. ये all these facilities are available at your institution.
Yes, ma'am. These are the available. We have done with the collaborative work with some different institute also.
Okay. So what is the outcome of this research?
Ma'am, we have the synthesized this membrane. We go into the surface morphology and we have checked the what is the BOD, COD, and the TDS. Out of that once we can check the BOD is reduced, COD is reduced,
Which
and
kind
the
of water सर? जो polluted water है, pond water है, real world water है, या किसकी बात कर रहे हैं आप?
मैम वेस्ट वाटर जो क्या कहते हैं हमारे इंस्टीट्यूट में जो वेस्ट वाटर प्रोड्यूस हो रहा था उसको हम लोगों ने लेके ट्रीट किया।
जैसे वेस्ट वाटर क्या हो गया? जैसे आपका ड्रेनेज का पानी हो गया, लैब का
ड्रेनेज पानी, ड्रेनेज वाटर में।
ड्रेनेज का पानी हो गया, तो इंस्टीट्यूट में क्या मतलब ड्रेनेज का पानी अगर होगा तो आपके क्या वहाँ रेजिडेंशियल कैंपस का भी मतलब डोमेस्टिक वेस्ट भी होगा
यस
उसमें?
मैम, यस मैम, रेजिडेंशियल कैंपस में।
रेजिडेंशियल कैंपस में आप वाटर ट्रीटमेंट तो दीज आर कैपेबल टू
They are
एब्जॉर्ब
they are also treating that waste water. We are also using the, we can treat this water with the help of the microbial fuel cell or not.
हाउ मच इट इज इफेक्टिव? कैसे आप इसके एक्सपेरिमेंटेशन आप कैसे रियल वर्ल्ड में करेंगे? जो मतलब आप वहां से वाटर ला करके लैब में उसकी एनालिसिस करेंगे?
यस मैम, उसकी नहीं, फाइनली जब हो जाएगा सेटअप देन वी गो place that one microbial fuel cell on the larger scale, then we can
the
treat
model of that
yeah, this is a prototype we are developing.
Prototype, exactly. So then you have to go for the patent also.
Yes ma'am, if they successful we can also reduce the other type of the wastewater treatment. In that once we go to the pharmaceutical wastewater, different type of the other type of the industry, then it will be successful, then we can go for that once patenting for
आपको आप किस एक्सटेंट तक ये कंफर्म है कि ये मतलब कितने परसेंट तक इस प्रॉब्लम को सॉल्व कर सकता है? जैसे मान लीजिए 100% तो नहीं कर
no
पाएगा।
ma'am, around the 40 to 50%, because the cost of that is the anodic that is the cathode, and the anode in that one that is disturbed, and that is the proton exchange membrane. After 4 to 5 cycles they give the less current and they have produces the less amount of the electricity.
Then what that water जब आप 40 से 50% उसको प्यूरिफाई कर पाएंगे तो वो वाटर कहाँ आप यूटिलाइज करेंगे? इरिगेशन के लिए करेंगे या ड्रिंकिंग के लिए?
इरिगेशन के लिए मैम, इरिगेशन के लिए, और क्या कहते हैं सीवेज उसमें सप्लाई के लिए,
सप्लाई के
इरिगेशन के लिए हम यूज कर
तो,
सकते हैं मैम।
तो आपके काम में जैसे क्या इस तरह के काम पहले भी हो चुके होंगे? क्या इनोवेशन है आपके काम
मैम
में?
वी आर सिंथेसाइज द न्यू टाइप ऑफ द प्रोटॉन एक्सचेंज मेम्ब्रेन।
Okay. You are doing very good job. And we are also doing phytoremediation with the help of water, the same kind of thing with the help of the plant. That's why
Yes,
I am
ma'am.
asking you about that कि आप भी उसी तरह का काम कर रहे हैं, बट आप माइक्रोबियल जो है वो क्या कहते हैं एमपीसी को लेके कर रहे हैं, मैग्नेटिक
MFC को
प्लांट
लेकर
को
कर रहे
लेकर के कर रहे हैं।
Yes,
तो
ma'am.
विश यू ऑल द बेस्ट फॉर योर
Thank you.
वर्क एंड आई अप्रिशिएट, इसको करते रहिए। अगर 50% भी समस्या को आप कम कर पा रहे हैं तो आप विजेता हैं। ठीक है, थैंक
Yes,
यू
ma'am.
वेरी
Okay. Okay. Thank you so much. Thank you so much.
Next one, please.
Please
Ma'am,
sum up your
Saraswati Chauhan.
presentation. Every candidate, I request you to please sum up your presentation within 5 minutes. Thank you very much.
Please join immediately. Am I audible?
Yes, ma'am.
कौन है ऑनलाइन? अपना नाम बताइए।
Please
Saraswati
share the
Chauhan, ma'am.
tell your name. Please share the screen and start
Saraswati
presentation.
Chauhan.
मैडम मैं इनके नाम लिख ले रही हूँ, आप मैच करा लीजिएगा।
हेलो
जी बताइए, जल्दी बताइए, हम आपकी आवाज सुन पा रहे हैं।
जी मैम।
We are having limited time. Okay, so you are having 5
ओके मैम।
minutes.
मैम
आप अपना इंट्रोडक्शन
स्क्रीन शेयर
बताइए।
की हूँ मैम, दिख रहा है?
नहीं दिख रहा है, आपका नाम केवल दिख रहा है।
सरस्वती चौहान।
Yes, ma'am.
आप बोलना शुरू करिए, स्क्रीन शेयर होता रहेगा।
ओके मैम।
आप अपना टाइटल बताइए,
Good
ऑब्जेक्टिव
evening.
बताइए, आउटकम
My
बताइए,
title
significance बताइए।
is Traditional Botanical Data Documentation from Raigad with reference to Uran tribe. My supervisor name is Dr. Deepak Kumar Soni sir, Dr. C.V. Raman University, Kargi Road, Kota, Bilaspur.
ऑब्जेक्टिव बताइए अपने काम का।
ओके मैम।
ऑब्जेक्टिव बताइए।
मैम Survey of economic plants used by the Uround tribes of the selected area, Raigad district, and herbarium preparation of the plant specimen collected, and identifications of herbarium prepare, and assessment of the economic earned by selling and plant study, and marketing, etc.
ठीक है, अब आउटकम बताइए, क्या आपके इस रिसर्च का क्या आउटकम हो सकता है और उससे सोसाइटी को क्या फायदा होगा?
मैम, इससे जो ट्रेडिशनल मेडिसिनल यूजेस पहले करते थे प्लांट्स वगैरह का, तो वो चीज अभी लगभग उतना ज्यादा मतलब डेवलप्ड नहीं हो पाया है। उसका डॉक्यूमेंटेशन करके हम प्रेजेंट कर सकते हैं, तो अपना जो कल्चरल वैल्यू है उसके अकॉर्डिंग, और जो प्लांट्स वगैरह है उसका जो बेनिफिशियल रोल है, वो सब चीज को हम बता सकते हैं। डॉक्यूमेंटेशन है वहां पर, हम लोग जितने प्लांट्स का स्टडी करके और
आप ये
जो
बताइए,
भी
जो
प्लांट,
हर्बेरियम शीट बनाएंगे उसका साइज क्या रखेंगे, स्टैंडर्ड साइज क्या रखेंगे?
सर मैम A4 साइज का रखूंगी मैम।
A4 साइज थोड़ा ना होता है, इसको जाके पढ़िएगा, ठीक है?
ओके मैम।
ठीक है, चलिए नेक्स्ट।
ओके, थैंक यू।
Next participant.
May I go next, ma'am?
हाँ हाँ,
Yes, tell your name.
This is
my
general
name
listen, this is general instruction for all the participants. Either you share your PPT. If you are not able to share your PPT, please turn on your video before presentation. Thank you.
Okay. My name is Nancy Jane. I am sharing my screen. Is it visible? And shall I proceed?
Yes, it is visible.
Okay, so my name is Nancy Jain. I'm a research scholar and I'm also an assistant professor. I am affiliated to Veer Narmad South Gujarat University. And my topic of presentation today is tolerance and pigment modulation in soil bacteria exposed to copper and lead stress. Now, as mentioned, since we are all in a time crunch, I'll just keep it short and go to the main points. So basically, I have worked on heavy metal contamination and how we can relate it to microbial pigment and whether they are having any effect on that pigment. So I have linked basically the metal tolerance and the pigment modulation in soil bacteria. Now the objectives are basically that I have isolated pigment producing bacteria from soil samples which is in and around Surat, and I have assessed the concentrations, varying concentrations of copper and lead towards them, and I have seen whether the pigment is getting changed or if there is a loss or if they are producing more pigment in case when we are giving them stress basically of copper or lead, and so that we can identify these specific microbes for bioremediation applications. That this is my rationale. So basically pigment modulation, whether it is happening and if microbes are growing towards the environmental stress or not, and if we can use them for a sustainable alternative or not. That was the rationale behind this whole work. So what I have done is I have collected sample from 34 different locations, and I have carried out 10-fold serial dilution and then spread
Madam,
film.
is it possible to collect the samples from 34 different locations?
Yes, ma'am. I have selected different locations, as in industrial locations, farm soils, different kinds of locations are there for 34, and from all of these different locations
34 का मतलब 34 locations means industrial, और आप क्या पूरा गुजरात को फोकस कर रही हैं?
नहीं, पूरा गुजरात नहीं मैम, सिर्फ सूरत या सूरत के आसपास। तो आई हैव उसमें भी मैंने अलग अलग काम किया है,
तो
जैसे
आप
फार्म
ट्रिप्लिकेट में कलेक्शन लेंगी या ट्रिप्लिकेट में
नहीं,
लेंगी तो
सॉइल
34
कलेक्शन ट्रिप्लिकेट्स में नहीं किया है। सॉइल कलेक्शन तो सिंगल ही किया है, बट उससे फिर ट्रिप्लिकेट्स में वर्क किया है।
अच्छा अच्छा, तो किस तरह का बैक्टीरिया ये ग्राम पॉजिटिव या नेगेटिव है जो कि पिगमेंटेशन को कंटेन करता है और सॉइल रेमेडिएशन का के लिए काम कर रहा है?
Ma'am, I've got various different isolates. I got around 90 isolates from these samples, and I have
the culture of these viable culture of these.
Yes, ma'am, viable. After I got the isolated colony, so I have streaked them. I've done the gram staining. I've noted the colony characteristics and everything. but
Then
I'm
you
not
have
in.
to go for the sequencing for the identification of these bacteria, which is found more effective.
Yeah, but I have narrowed it down. Like this is actually a part of my PhD work. So
Okay.
yeah, so this is one of the work that I am.
कैरेक्टराइजेशन के आधार पे बस खत्म कर देंगे, उसके सीक्वेंसिंग वगैरह पे नहीं जाएंगी?
नहीं, सीक्वेंसिंग पे जाएंगे मैम, बट जब नैरो डाउन कर देंगे ना उसे, मतलब 90 के 90 को तो आई कैन नॉट सीक्वेंस, तो मे बी उसमें से 5 या 6 को जब नैरो डाउन कर देंगे जो इफेक्टिव है, तो उसका पूरा ऑप्टिमाइजेशन, उसका सीक्वेंसिंग और आगे का
तो
जो
आप उन
एप्लीकेशन।
सॉइल को फिर क्या उनको आप सॉइल में जो आप कंटामिनेटेड सॉइल लेंगे उसमें उनके इफेक्ट को देखेंगे?
यस मैम।
सॉइल टेस्टिंग करेंगे कि इसमें लेड को कितना और क्या, दो कौन कौन चीजों पे कौन सी है, कौन से आपके हैं वो लेड
लेड
और
और कॉपर है मैम।
कॉपर हैं कि ये कम हो रहे हैं कि नहीं कम हो रहे हैं?
एग्जैक्टली।
एग्जैक्टली आपकी मेथोडोलॉजी ये होगी और आपका आउटकम क्या होगा, उसका सिग्निफिकेंस क्या होगा। आप अच्छा काम कर रही हैं, बिकॉज इंडिया का कोई ऐसा सॉइल नहीं है जिसमें कि कंटामिनेशन ना हो। इट्स
ना
वेरी
हो।
नेसेसरी
यस।
टू रेमिडिएट द सॉइल। क्यों? क्योंकि जब
यस मैम।
सॉइल में अगर कंटामिनेशन होगा, ये हैवी मेटल्स होंगे, तो ये हमारे हमारे जो है बॉडी में, फूड में, एयर में, हर
हर
जगह ये
जगह
सर्कुलेट
एंटर होते
करेंगे।
हैं, बायो मैग्नीफाई होते हैं।
तो मैं आपकी तारीफ करती हूँ ऐसा सब्जेक्ट लेने पर, लेकिन आप मेहनत से काम करिए, क्योंकि 34
यस
सॉइल
मैम।
सैंपल से ना मेरे दिमाग में ये घूम रहा है कि आजकल के बच्चे तो काम को, चीजें को छोटा करना चाहते हैं। 34 सॉइल सैंपल कलेक्ट करना और 90 के आसपास आइसोलेट्स को मेंटेन करना, इट्स वेरी डिफिकल्ट। प्योर कल्चर करना और उसको मेंटेन करके रखना, ये थोड़ा
यस
मुश्किल। अगर
मैम।
आप ऐसा कर रही हैं तो वाकई काबिल तारीफ है। मैं
यस मैम।
आपको इसके लिए शुभकामना देती हूँ।
थैंक यू।
इट्स गुड फॉर यू। ओके, वेलकम। आप ऑफलाइन आप जा सकती हैं। आप जो है वो
नेक्स्ट कैंडिडेट प्लीज।
ओके मैम।
नेक्स्ट कैंडिडेट प्लीज प्रेजेंट हियर।
Hello, ma'am.
Yes, please start, ma'am. Tell me your name and start.
Myself, Manisha Deri. I am a research scholar.
Manisha.
My research center is Government Bilasa College, Bilaspur. My presentation, I am start sharing my screen. My my presentation topic is role of ant microbe interaction in contaminated soil.
Ma'am, please don't focus on the sharing. Please give your presentation, ma'am.
Okay, yes, ma'am.
सॉइल कंटामिनेशन से आप क्या समझती हैं?
सॉइल कंटामिनेशन मीन्स आजकल जो भी सॉइल में पॉल्यूशन हो रहा है, केमिकल।
वो कहाँ से रहा है पोल्यूशन?
केमिकल केमिकल यूज के कारण, और जो भी खेतों में हम लोग फर्टिलाइजर यूज करते हैं, और सॉइल इरोशन।
ऑब्जेक्टिव क्या है आपकी स्टडी का?
हेलो मैम।
ऑब्जेक्टिव बताइए अपने काम का।
जो भी सॉइल सॉइल में जो कंटामिनेशन है, उसको हम लोग बायो रेमिडिएशन का यूज करके सॉइल को प्योर करना और उसको उपयोगी बनाना।
आपके साइट्स कौन कौन से हैं?
मैम, ये मेरा रिसर्च टॉपिक नहीं है, मेरा रिसर्च टॉपिक कुछ दूसरा है मैम, तो ये उससे रिलेटेड टॉपिक है।
अच्छा, तो आप क्या रिव्यू पेपर दे रही हैं कि आप रिसर्च पेपर प्रेजेंट कर रही हैं?
ये दुकान वाले को देख रहे हो क्या? नहीं।
Next candidate, please.
हेलो मैम।
Yes, please. Next candidate, please.
हेलो मैम।
Please present yourself.
हेलो। हेलो।
Sir, please tell me your name.
Anant Mehta, please start your video.
Ma'am, can I present myself?
Wait for a while.
Hello.
Gauri, wait for a while. First, Anant Mehta.
Okay, sir.
Yes, Anand ji, please proceed.
जी। Good evening, respected teachers and everyone present here. Today I am going to present my research work titled Development of an IoT Integrated Machine Learning Framework for Precision Agriculture Resource Optimization and Plants Diversity Conservation. This study is carried out by me, Anant Kumar Matha, under the guidance of Dr. Rahul Kumar Pandey from C.V. Raman University. Abstract. In this research, we propose a smart agriculture system that integrates IoT-based sensor with machine learning models. IoT sensor continuously monitoring soil moisture, pH, thermometer, humidity, and other environment conditions. This real-time data is analyzed using advanced machine learning algorithm to predict crop needs, optimize resource, and detect decrease early. The system also supports biodiversity conservation, making agriculture both produce and sustainability. Agriculture today faces challenges like climate variability, soil.
आनंद जी मेरी बात सुनिए। आप
Yes.
ऑब्जेक्टिव बताइए अपने 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 टाइटल का, अपने रिसर्च वर्क का। आप किस डिपार्टमेंट से हैं? कंप्यूटर साइंस से हैं?
जी मैम, मैं कंप्यूटर साइंस से हूँ।
जी। तो आप ऑब्जेक्टिव बताइए, बहुत प्रिसाइज होइए अपने काम के ऊपर। इंट्रोडक्शन पढ़ने की जरूरत नहीं है।
जी मैम।
क्या आपके काम का क्या ऑब्जेक्टिव है, उसका आउटकम क्या है और उसका सिग्निफिकेंस क्या है, ये 2 मिनट में बताइए।
इसमें ये है मैम कि ये जो रायगढ़ क्षेत्र में जैसा हम लोग देख रहे हैं, जो किसानी यहाँ से होते हैं, तो इसमें भी बहुत सारे वो ट्रेडिशनल वाला चल रहा है। तो उसमें यहाँ पे हम लोग मैम वो जो सेंसर होते हैं, जिसके बेस पे सारी चीज का ड्रोन के माध्यम से मॉनिटरिंग करें और जो खेती है उसको अच्छे से करें। और उसमें आपके बहुत सारे, जैसे अगर हम वाटर का भी देखें तो बहुत सारे वेस्ट हो जाते हैं।
तो ये
तो
सेंसर आप बनाएंगे?
नहीं
ये
मैम,
सेंसर
बना हुआ को इधर यूज करेंगे। ऑलरेडी इसमें तो बहुत सारे सेंसर अभी बन चुका है।
अच्छा, तो वो
जी।
बना हुआ सेंसर आप क्या मार्केट से लेंगे, आपके सुपरवाइजर के पास है?
नहीं, मार्केट से मैम।
तो
उसको यहाँ
मार्केट
पे हाँ, वहाँ उसको यहाँ पे इम्प्लीमेंट करेंगे मैम, जिसमें अब यहाँ पे देखेंगे तो यहाँ पे वैसे कुछ मशीनरी नहीं है, वो ट्रेडिशनल ही सब चल रहा है।
तो आप क्या, जैसे कि पानी की जरूरत है तो उससे लगा के जानेंगे कि अभी इसको इरिगेशन की जरूरत है, इसको पेस्टिसाइड की जरूरत है, इसको
जी
मतलब
जी
इंसेक्टिसाइड की
मैम। हाँ
जरूरत है, ये
वो सब
आप
मॉनिटरिंग करेगा। जी मैम।
अच्छा, तो मतलब एग्रीकल्चर को बेहतर बनाने के लिए, तो इसमें
जी
इनोवेशन क्या हो गया? मतलब जैसे रिसर्च का मतलब तो इनोवेशन होता है। मार्केट में डिवाइस मौजूद है, वो तो कोई भी, जैसे मान लीजिए मिक्सी बनी हुई है, कोई भी
जी।
उसको ले जाकर के उसमें कुछ भी पीस सकता है। इसमें इनोवेशन क्या है आपके काम
अभी
में? इनोवेशन क्या, इनोवेशन का मतलब समझते हैं?
जी मैम, अभी रियल टाइम जो डेटा है ना मैम, वो रियल टाइम डेटा नहीं मिल पाता है। मतलब
अच्छा।
उतना जो डेटाबेस है अभी उतना स्ट्रांग नहीं है। बाकी सेंसर तो है, लेकिन उसको डेटाबेस से उतना अच्छे से अभी इम्प्लीमेंट नहीं हो पाया है।
अच्छा, फिर
तो
तो आप
रियल टाइम,
अच्छे से इम्प्लीमेंट करेंगे उसको?
जी, रियल टाइम डेटा जैसे मिल सके।
तो आप कितनी जगह पे करेंगे? आपके लोकेशन क्या क्या होंगे?
ये लोकेशन तो मैम, यहाँ पे मैं पटेल पाली में रहता हूँ, तो पटेल पाली का उसमें लिखा भी हूँ।
तो आप कौन कौन सी सीजन में करेंगे इसको, रबी, खरीफ, या किस सीजन में, या किस क्रॉप के ऊपर करेंगे?
उसको तो अभी नहीं किया है।
नहीं किए हैं? तो आपकी मेथोडोलॉजी आपको पता होनी चाहिए ना, कि जब आप रिसर्च सिनॉप्सिस लिखते हैं तो आपको मेथोडोलॉजी पता होनी चाहिए कि आप रबी के सीजन में करेंगे, खरीफ के सीजन में करेंगे, तब ना सीजनल आप कुछ डेटा आप कलेक्ट कर पाएंगे।
जी
और
मैम।
जब भी रिसर्च करते हैं तो वो हमेशा इनोवेशन बेस्ड होता है। इनोवेशन का मतलब उसमें नवीनता क्या है।
जी
ठीक है,
जी
मैं ये नहीं
जी
कह रही हूँ कि आप कोई चीज इजाद करिए, लेकिन उसमें कुछ नया एंगल तो बताइए हमको कि आप इस नए एंगल से करेंगे। मान लीजिए मशीन है, टीवी है, किसी ने बना दी, हम ला करके उसको कहीं लगा दे, चाहे बेडरूम में लगा दे, ड्राइंग रूम में लगा दे, वो चलेगी। उसमें नवीनता क्या है बताइए।
जी
सुपरवाइजर से जाइएगा तो अपने पूछिएगा, ठीक है ना, कि सर इसमें क्या नवीनता है, क्या सीजनल वेरिएशन हमको देखना है, सीजन वाइज देखना है, कैसे देखना है, मेथोडोलॉजी से डिस्कस कीजिएगा। ठीक है, धन्यवाद। चलिए नेक्स्ट।
Next, next, please. Next participant, please clear your screen and start the presentation.
हेलो
वो मैडम कोई बता रही थी ना।
हेलो एक्सक्यूज मी मैम।
जी जी, क्या नाम है
मेरा
आपका?
नाम उपासना मेश्राम है।
जी जी, बताइए।
जी मैम, मैं क्या अपना प्रेजेंटेशन स्टार्ट कर सकती हूँ?
जी जी, स्टार्ट करिए।
It's okay.
मैम थोड़ा सा
First,
ना
first
स्लाइड शो करने
start
में
your
प्रॉब्लम
video.
आ रहा है, कुछ यहाँ दिक्कत है, तो
It's
मैं
okay.
डायरेक्ट ऐसे ही प्रेजेंट कर सकती हूँ क्या?
Listen, Upasana, please turn on your video and proceed for the presentation. Very simple.
ओके मैम।
क्या नाम है इनका? उपासना।
Upasana, unmute yourself. You are visible. Just unmute yourself and start your presentation.
धन्यवाद सर। मेरा नाम उपासना मेश्राम है। मैं शोधार्थी अटल बिहारी वाजपेयी यूनिवर्सिटी से हूँ। मैं शिक्षा विभाग की शोधार्थी हूँ। मेरा विषय है स्कूली छात्रों में समालोचनात्मक चिंतन को विकसित करने में पर्यावरणीय साक्षरता की भूमिका। इसमें मेरा जो उद्देश्य है, विद्यालयों में पर्यावरणीय शिक्षा की स्थिति, स्वरूप और महत्व का विश्लेषण करना, विशेषकर यह दिखाना कि स्कूलों में कौन कौन से क्रियाकलाप पाठ्यक्रम पर्यावरणीय चेतना बढ़ाने हेतु उपयोग किए जा रहे हैं। साथ ही यह समझना कि क्या विद्यालयों में पर्यावरणीय शिक्षा केवल सफाई वृक्षारोपण तक सीमित है या उसे पाठ्यक्रम और व्यवहारिक गतिविधियों के साथ भी समेकित किया जा सकता है। इसमें मेरी जो शोध विधि है वो है साहित्य संदर्भों और विद्यालयों में जो
मैडम
प्रचलित
मैडम
गतिविधि।
एक मिनट, मैडम एक बात बताइए, स्कूल में आप मतलब कहाँ से कहाँ तक के बच्चों को टारगेट कर रही हैं? क्लास 1 से
मैम
क्लास
मैं
8 तक?
ये हायर सेकेंडरी स्कूल, मिडिल और हायर सेकेंडरी स्कूल के जो बच्चे हैं उनको लूंगी।
12 तक।
हेलो
अच्छा हाँ, आप बताइए, ये बताइए कि आप जो है वो मतलब 6 से लेकर 12 तक को आप कर रही हैं फोकस?
जी जी जी।
अच्छा अच्छा, बताइए अपना अपने काम का आप उद्देश्य, आउटकम बताइए।
ओके मैम।
आउटकम का मतलब है कि उससे आपको क्या चीज मिलेगी।
क्या कंक्लूजन? जी मैम
जी
ठीक है। ये मैम एक्चुअल में इसमें मैंने टारगेट किया है बच्चों को, परंतु इसके लिए मैंने ये मेन है सेकेंडरी डाटाबेस पे रहेगा मेरा। सेकेंडरी डाटाबेस पे है ये जो मेरा रहेगा मेथोडोलॉजी उस पे। इसका मैंने निष्कर्ष यदि देखती हूँ तो जो
सेकेंडरी
शोध
डेटाबेस
पत्र
क्या होता है?
मैम, जो मैं सेकेंडरी डेटाबेस मतलब जो मैंने जैसे यूनेस्को का एक रिपोर्ट है, उस रिपोर्ट के अनुसार मैं अपना ये ले रही हूँ। जितने भी उससे पहले जितने भी रिव्यूज होंगे उस लिटरेचर से संबंधित, उन लिटरेचर्स को मैं रीड करके फिर अपना एनालिसिस बताऊंगी। उस पे बच्चों का जैसे डाटा है, मेरे मेन थीसिस का अलग है, इसका अलग है मैम।
तो मैडम आप जो है वो आप डेटा कलेक्शन नहीं करेंगे स्कूलों में जा करके?
नहीं नहीं मैम, इसका डेटा कलेक्शन नहीं करूंगी मैं। ये तो सेकेंडरी डाटाबेस रहेगा, एनालिसिस पे रहेगा।
आप बस बैठ के स्टैटिस्टिकल एनोवा करेंगी या एसपीएसएस
जी
पे एनालिसिस करेंगी? एसपीएसएस पे?
जी जी।
अच्छा ठीक है, तो इसका सिग्निफिकेंस, महत्व क्या होगा?
मैम महत्व यदि मैं देखती हूँ इस पे तो जितने भी आपके
अच्छा हमको
पर्यावरण।
दो शब्दों में ये बताइए कि यूनेस्को का डेटाबेस क्या कहता कहता है, जो प्राइमरी डेटा है वो क्या कहता है कि बच्चों के बीच, इन्वायरमेंटल स्टडीज के बीच कुछ अवेयरनेस है, वो कुछ करते धरते हैं या उनके टीचर्स उनको कुछ बताते हैं, ऐसा कुछ लिखा है?
जी मैम, उसमें उन्होंने यह स्पष्ट किया है कि पर्यावरण शिक्षा का उद्देश्य केवल जानकारी देना नहीं है, बल्कि ऐसे व्यवहार और दृष्टिकोण विकसित करना है जो पारिस्थितिकी तंत्र जो है हमारे
ये
इकोसिस्टम
दुनिया,
उसकी सुरक्षा।
हाँ, ये दुनिया में हर आदमी जानता है, तो क्या ये एक्चुअली हो रहा है? उनके व्यक्तित्व में ये इनकलकेट हो रहा है? उनके व्यक्तित्व में ये बातें जा रही हैं स्कूली शिक्षा से?
मैम देखिए, डेटा के अनुसार अगर मैं एनालिसिस कर रही हूँ तो वो तो जा रहा है, परंतु अगर हम एक्चुअली देखेंगे तो उस पे बहुत कम होता है मैम। ये आप भी जान रही हो और
नहीं
मैं
है,
भी जान रही हूँ, परंतु
प्रैक्टिस
हमको
में नहीं है,
अवेयर करना पड़ेगा बच्चों को।
प्रैक्टिस में नहीं है।
जी जी जी।
प्रैक्टिस में नहीं। ठीक है मैडम, धन्यवाद बहुत बहुत, आपका काम बहुत अच्छा है, मन लगा के करिए। लेकिन मैं ये कहूंगी कि आप अपने लोकल स्कूलों में भी जा करके कुछ अब पूछिए कि भाई आप लोग ये करा रहे हैं कि नहीं। उनके एक्चुअली हमें उन्हें उनके व्यक्तित्व में इनकलकेट करना है। मैं भी क्या कहते हैं कि हायर एजुकेशन में नेचुरल रिसोर्स एंड मैनेजमेंट की क्लासेस लेती हूँ, इसलिए मैं पर्सनली आपको बोल रही हूँ कि ये चीजें हर व्यक्ति के, वन एंड ऑल इंडिविजुअल के जो हमारी पर्सनालिटी, हमारा व्यक्तित्व है, हमारी आदतें हैं, उसमें शामिल होना चाहिए। किताबों में तो बहुत सारी चीजें लिखी हुई है, रामचरितमानस
यस
में
मैम, ये बिल्कुल सही चीजें हैं। हमें बच्चों को अवेयर करना चाहिए, अपने आसपास के लोगों को अवेयर करना चाहिए।
थैंक यू मैडम,
थैंक यू
थैंक यू
सो
वेरी
मच
मच।
मैम।
वी आर हैविंग वेरी शॉर्ट ऑफ टाइम। ओके,
जी जी,
थैंक यू।
थैंक यू सो मच मैम।
Next participant. Anyone there? Anyone?
Ma'am, can I speak, ma'am? This is Priyanka.
जी बोलिए, डॉक्टर प्रियंका।
Yes, ma'am. Yes, ma'am.
पूरा
Dr.
क्या नाम है
Priyanka.
आपका?
Ma'am, Dr. M.S. Priyanka Yadav.
जी बोलिए।
Yes, ma'am. I am Dr. M.S. Priyanka Yadav, working as Assistant Manager in SSCL HQ Bilaspur, and my co-authors are Shri Praveen Kumar, General Manager Mining, SSCL HQ, and George K. Verghese, my guide, NIT Calicut, and Amit Verma, Deputy Manager, SSCL HQ, ma'am. Ma'am, I just don't want to take a lot of time. I understand we are running out of time. My paper is is innovations and paradigm shift in the contaminants of emerging concern, that is sulfonamides. So we all are talking about the contamination, pollution in soil. हमारे इस बीच में there is something else. Mostly my objective is mostly on a review paper and also on the treatment aspect as well. As of now, I would like to tell the review aspect. We all know that we generate wastewater, and wastewater is mostly of organic content. So अभी जो हो रहा है मतलब
We are not able to hear you. We are not able to hear you.
5% more than what usually it happened before 2000s, and the rate it has increased was 103% incremental. So this shows the abruptly increase, inappropriate and abuse of antibiotics is happening in India. Ultimately, where we are consuming antibiotics, and we know antibiotics are.
What is the objective of your study?
Ma'am, this is mostly a review paper where I would where I'm
Review का भी
concentrating.
objective होता है मैडम?
Yes, ma'am. I'm coming to the point.
Okay.
Yeah, objective ये है मैम, हमारा जो एंटीबायोटिक्स हम कंज्यूम करते हैं, that is being digested in our system, that is our human body, and the antibiotics are also used in the animals. So both are metabolic systems. So वो digest होके that are coming as a secondary products. हम tetracyclines लेते हैं and azithromycins लेते हैं, but when we digest and come it out as urine and excreta, their remnants are still present, and these chemical compounds cannot be treated in the existing STPs or ETPs, mostly STPs. So when the STPs, we have two to three types of treatment, that is primary, secondary and tertiary. तो secondary treatment में it is mostly aerobic.
मैडम आप कहना क्या चाहती हैं, आप कहना क्या चाहती हैं?
I would like to tell that these antibiotics are not considered as the contaminants, not taken seriously. But
ऐसे आप कैसे कह सकती हैं?
no, ma'am.
एंटीबायोटिक के ऊपर
Yes,
जो
yes.
है
No. See,
हो
not
रही है।
taken seriously in the sense, ma'am. See, now the present existing STPs don't have the capacity to treat these
वाव का
antibiotics,
नाम
ma'am.
सुना है आपने? WHO में वाव का नाम सुना हुआ है आपने?
कौन सा मैम?
वाव एक मुहिम चलती है।
Okay.
वाव के हिसाब से WHO वर्ल्ड हेल्थ ऑर्गेनाइजेशन जो है, जो एंटीबायोटिक के रेजिस्टेंस पे बात करती है।
Exactly, ma'am. I'm coming to that point
हाँ,
only.
तो आइए ना, उस पे आइए ना, आप
उसमें
भूमिका मत बांधिए, आप अपने काम पर आइए। हम लोग इतना जानकार हैं कि आप किस पे बात करना चाहते हैं।
Okay. Yes, fine, ma'am. See, now mine is mostly review paper. I am trying to find the papers where these antibiotics are being detected in different compartments of environment, be it be water,
मैडम
soil.
देखिए, एंटीबायोटिक रेजिस्टेंस बहुत बड़ा प्रॉब्लम है। और एक मैं आपको बैक्टीरिया का नाम भी बता देती हूँ, स्टेफाइलोकोकस, जो कि जो है वो जो बच्चे क्या कहते हैं, NICU में होते हैं, जो कि बहुत कम समय पे पैदा हो जाते हैं, उनके अंदर जो न्यूमोनिया मतलब इस तरह के प्रॉब्लम्स क्रिएट करते हैं, वो रेजिस्टेंट हो चुके हैं और उनकी डेथ हो जा रही। तो वी आर वेरी मच एक्वेंटेड अबाउट द severe effect of the antibiotic resistance.
Ma'am,
ठीक है?
I'm not a microbiologist, so I cannot comment about the types of microbial resistance. I'm just trying to tell the current existing STPs who are actually dependent on microbes to degrade the organic matter cannot perform the activity of degrading these chemical compounds. These are chemical compounds, antibiotics. So उनका ताकत नहीं है इसका breakdown करने के लिए. So we need a different treatments, definitely. Okay, my objective is finding the places where the antibiotics are being detected naturally. अगर आप कहीं भी हमारा river का भी पानी आप sampling करेंगे, we can find directly the concentrations ranging from 0.1 milligram per liter to 19 milligram per liter. My friends and we have conducted some study in Chennai and the Mumbai. We have found so many, approximately 98 antibiotics metabolites in the wastewater and also the flowing water, river waters. यहाँ चेन्नई में हमारा अडियार river होती है, तो we are able to find randomly any sample you grab it, you can find some antibiotics. So my objective was to find where and all these being detected.
Let me know the outcome, conclusions. Come on the conclusions.
Conclusions is, ma'am, different types of methodologies to detect. अभी जो metabolites है, ma'am, हमारा metabolites है, they don't have a methodology in HPLC or LCMS or GCMS. So हम different methodology explore करते हैं ताकि वो उसको detect कर पाए. This is one objective, and second objective is treatment methods. Treatment methods like AOPs, that is advanced oxidation processes, using photocatalysis, nanomaterials. हमारा proposal है next phase के लिए. So this is our work, ma'am.
Thank you very much. Thank you.
Thank
you. You can leave.
Yes, ma'am. Thank you.
Hello.
Next one, please.
Next participant.
Ma'am, it's Gauri. Can I present?
Yes, you can continue.
Okay,
गौरी, बहुत शॉर्ट में 2 मिनट में अपना काम का ऑब्जेक्टिव बताइए, चाहे हिंदी अंग्रेजी में पढ़ने की जरूरत नहीं है। आउटकम बताइए और उसका सिग्निफिकेंस, महत्व बताइए। स्लाइड पढ़ने की जरूरत नहीं है बेटा।
Okay, ma'am. Ma'am, wait a minute, just a second. Ma'am, I'm working on exopolysaccharide production in cocoid bacteria, cyanobacteria, that's Cynococcus species. And hello? Ma'am, am I audible? Yes, ma'am. I'm a PhD scholar from CCS University Meerut, and and this presentation I'm presenting. I worked on different phosphate concentration and uska effect on exopolysaccharide production. I have taken as a BG11 as my control, and in which K2HPO4 is taken as a phosphate source. So I have taken a different concentration
What is the objective of your study?
Ma'am, to standardize the conditions in which exopolysaccharide from cyanobacteria could be enhanced.
Mm-hmm.
So
What
I
is the
have,
outcome?
ma'am, I have tried a different concentration of phosphate K2HPO4, and I find if the standard condition that is mentioned in BG11, if we take the only 75% phosphate concentration for this Sinococcus strain, then the EPS concentration would be enhanced.
Okay. What is the significance of this? दो
Ma'am,
लाइन में बताइए.
Ma'am, as the exopolysaccharides could be used as a bioplastic, it could be used in various cosmetic formulation, like in it has the Ma and cytonomin, which is UV protectant. So if we enhance these, if we able to standardize the conditions, so it would be beneficial.
Okay. You are doing nice work. Keep it up. Okay. You can leave.
Okay, thank you, ma'am.
Next participant, please summarize your presentation within 2 minutes.
Shailesh, we can see you. Go through.
ऑब्जेक्टिव बताइए अपना. व्हाट इज योर ऑब्जेक्टिव? अभी कितने स्टूडेंट हैं? नहीं हो रहा है. क्या करें?